Buenas tardes, bienvenidos a la sesión de la tarde. En este momento entonces nos alistamos para dar inicio a la segunda parte de la cátedra, que consiste en un taller. En primera instancia voy a presentarles a los conferencistas que estarán con ustedes durante esta parte de la cátedra. Estarán con ustedes el profesor Kim Jordan, a quien ustedes ya conocen. En algún momento el profesor Leonardo Mariño también se nos va a unir. El profesor Mariño es investigador en epidemiología y genética en el National Institute of Minority Health and Health Disparities de los Estados Unidos y realizó un doctorado en bioquímica en la Universidad de Texas A&M en Texas y recibiendo apoyo a través de una beca Fulbright con ciencias en el año 1997. Es el más antiguo de los ex-Fulbrighters que estamos hoy reunidos. Los doctores Jordan y Mariño van a estar acompañados por Shivam Pharma. Shivam Pharma trabaja como bioinformático en el laboratorio del doctor Leonardo Mariño en el National Institute of Minority Health and Health Disparities en los Estados Unidos. Ellos van a hacer una serie de demostraciones, van a explicar la metodología de todo lo que ustedes vieron en la mañana. En primera instancia quiero avisarles que mucho de lo que se va a realizar dentro del taller va a estar hablado en inglés, pues digamos esto por los instructores que tenemos. Vamos a realizar una serie de ejercicios guiados que ustedes pueden realizar de manera simultánea de sus computadores o simplemente estar atentos al desarrollo y posteriormente en el material que va a estar disponible después de la cátedra, los pueden tratar de emular, de reconstruir y de volver a realizar. Entonces vamos a hacer 6 ejercicios. En primera instancia vamos a hacer un ejercicio de caracterización genómica y formato de los datos que se tienen que, cómo deben estar los datos pues para poder hacer análisis. Luego hacemos 2 sesiones de ancestría genética. En la primera vamos a hacer un análisis de componentes principales y en la segunda parte un análisis de mestizaje genómico, así lo hemos denominado. Esto es una traducción de admixture, simplemente como mestizaje genómico, lo hemos traducido al español. La cuarta sesión será sobre diagnóstico genómico de enfermedades raras. En la quinta vamos a ver cálculo de riesgo poligénico para enfermedades comunes y complejas, son como los dos extremos. En la sesión 4 hemos aplicado pues el diagnóstico a enfermedades raras, también a veces llamadas mendelianas o huérfanas. Y al otro extremo tenemos el cálculo de riesgo poligénico para enfermedades que son comunes y complejas. Y finalizamos con un taller sobre farmacogenómica para el tratamiento de enfermedades. Entonces, en este caso, pues la idea es que ustedes, a medida que va avanzando la demostración, vayan haciendo las preguntas por el chat y estas serán resueltas en el momento pues, más apropiado por los profesores y los instructores del ejercicio. No siendo más, entonces le doy la palabra a el profesor Jordan y a Shivam para que adelanten entonces el taller. Bienvenidos.
Okay, good. Thank you, Augusto. So from here forward, we will present in English, and I'm going to get started. I'm just going to give an overview of what we're going to do, and then I'm going to turn it over to Shivam. And Shivam is really going to do most of the hard work this afternoon. So the first thing I want to do is introduce everyone to this web page, this GitHub page, where all the materials for today's exercise can be found. So this is the URL, this is the website for the page, and you can find this link in the chat for all of the platforms that we're using. So for those of you who are watching me live right now, I'm just going to give you the opportunity to get this link, click on this link from the chat and go to the page. And again, if you can't type it or see it from here, it's available in the chat for all of the platform. And this web page will show as I show you the page. So I'm going to go ahead and move on to that page. And again, the URL is here. So this page is open to the public and you should all have access to it. So what are we going to do today? So you heard from Augusto that we're going to go over a series of 6 different areas of essentially computational genomics or bioinformatics in support of the kind of analyses that you will need to do precision public health or precision medicine. Now we have, thanks to the hard work of Shashwat, who you heard about, and Shivam, organized a lot of material for you, maybe more than we can cover in 3 hours. And that's why I want to show this page first. This page has everything you need to do all of this work on your own asynchronously after this class finishes. So there are 3 different things that you'll need. So all of the teaching is going to be done in Jupyter notebooks. And Shivam, when you get started, if you could just introduce the concept briefly of Jupyter notebooks. So all of these files, which end with IPYNB, are the Jupyter notebooks. And Shivam, again, if you want to go over this more as you get started, that's perfectly fine. There's a README file which explains what you need to do. So here's the README file, and the repository contains all the Jupyter notebooks. This tells you about all of the programs, what we call dependencies. So this is how you install Jupyter. This is how you install these different programs, Plink, R, etc., that you'll need to do the analyses, okay? And this tells you a little bit about the notebooks, and you can also get all of the data in the data file. So again, what's the idea? The idea here is you can watch us today demonstrate these tools synchronously or asynchronously later, but when and if you are ready and interested to go back and try some of these analyses on your own, you will have all of the programs that you need, or access to all of the programs that you need to install before you get started, all of the Jupyter notebooks, each one of which corresponds to a specific exercise, and all of the data, each data set, each genomic data set corresponding to a specific exercise. So that's everything you will need to do this on your own. And again, here's the URL. This is on GitHub. It's publicly available, so it should be accessible to everyone. And that link is available in the chats of all of the platforms where this conference is being hosted. Okay? Now, the way we're going to do each one of these sessions, so you just heard from Augusto, the first one is characterization, genomic characterization in data formats. Then we will go to genetic ancestry for 2 sessions, one on principal components analysis, one on admixture, then genomic diagnosis of rare diseases, polygenic risk scores, and pharmacogenomics. So for each one of these sessions, we'll start with a high-level overview of the concepts that I will give in a PowerPoint presentation, and we will make all these presentations available as well on the GitHub page. And then Shivam will go through the exercise step by step in the Jupyter notebook. If you don't know what a Jupyter notebook is, Shivam will introduce that concept as he gets started on the first exercise. So with that, let's get started with the first concept, which is characterization of genome sequences and data formats. Now again, what I'm going to try to do briefly at the beginning of each of these 6 sessions is introduce the concepts at a high level leaving you to fill in the details if you want to come back asynchronously. So I will touch on what I think is the most important concept and leave the rest for you to go into. Okay, so the unit of study for precision public health and for precision medicine, at least with respect to what we're doing here today, is the single nucleotide variant. So positions in the human genome sequence that differ between individuals. And here we're showing an example of a single nucleotide variant, or an SNV. And the alleles are just different versions, or alelos in Espanol, are different nucleotide variants that exist at that position. So each single nucleotide variant is a single position in the genome, and it can have different alleles or different nucleotides at that position. And precision public health, genomic medicine, precision medicine are all about the relationship between these genetic variants and health and the response to medicine. Now, of course, you're probably aware that there's all kinds of different genomic variants, but here we're only talking about single nucleotide variants. It does not mean that the others are not important. It's just that most work in this field, at least to date, is done on single nucleotide variants. Now, if you have a single patient or an individual, how is it that you characterize their genome? And by characterize the genome, I simply mean identify all of the variants that they have around the genome. And there are three main technologies for doing this. The first one is genotyping using SNP arrays. This is the least expensive. The problem is you only get a small fraction of the genome, and you only look at positions where you already know there are variants. The next one is whole exome sequencing, where you only sequence the protein coding portion of the genome. It's a bit more expensive. It's less of the genome, but it generates a lot more data. You have to do more analysis, and it tells you about rare variants, and it focuses on the part of the genome that we have good reason to think is most important for health outcomes. And then finally, the most comprehensive is whole genome sequencing, where, as it sounds, you sequence the whole genome. This is much, much cheaper than it used to be, but it's still relatively expensive per sample. And there's a very high bioinformatics overhead in terms of the amount of analysis you have to do, but you gain the most information here. So I have provided information about how these techniques work, but I'm not going to go into that because of time here. But again, you will have access to these slides, and if you want more information, you can look there. What's most important with respect to what Shivam will do is how is it that you represent this variation, single nucleotide polymorphisms, as I show here, or single nucleotide variants. Computationally, how do we represent these data so that we can analyze them bioinformatically? And for any given single nucleotide variant or single nucleotide polymorphism, we often use those terms interchangeably. You show the chromosome location, the number, the position on the chromosome, and this is specified for what we call a specific genome version. There are different versions of the human genome reference sequence, and the chromosomal location is expressed in the context of a specific version of the human chromosome sequence. And you have an identifier, which is often an RSID, and then you show information about the variants. And the most common format is called the variant VCF, variant column format, okay? And you can look here on this Wikipedia page, and it tells you all about that format, but Shivam is going to tell you about that too. And so here are links to additional resources about genomic technologies, and here's additional resources about variant column. So with that, I will turn it over to Shivam for the demonstration part of things. So Shivam, take it away, and when you start this particular session, if you could just give a little bit of background on what a Jupyter Notebook is. And I know that we've already covered a lot of ground with respect to the GitHub page and the resources that are available. But if I missed anything there, by all means, you can talk about that. So Shivam, please take it away.
Okay. Professor Jordan, I think you're sharing the screen, so you'll have to unshare it before I can start. Thank you. All right, thank you, Professor Jordan, for giving us a brief introduction of how genotyping data and genomic data in completeness is stored. I will begin with giving a brief overview of what a Jupyter notebook is. So essentially, it's an open source software, and it's pretty much like any other text editor that you will come across, except it lets you dynamically compute programming languages and the code snippets you write. So for example, whenever you have a Jupyter notebook, the software open on your web browser, you'll have multiple options here. You can have Python 3, R based, or you can also run a native Linux based terminal. And you can write different pieces of codes, and I'll go more into it. So let's say we start a notebook with Python 3. So Python 3 is an open source language, and it's widely used among data scientists and bioinformaticians. So let's look at this little Jupyter notebook that we have here. So what Jupyter notebook lets you do is it provides you with these cells. So you can write any piece of code in these cells, and you can compute that code individually. So for example, print hello is a command in Python which lets you print hello on the terminal screen. So if I compute this command, Jupyter notebook only computes this particular cell, the first cell you see on your screen. Then I define a variable, a equals Jupyter. So what Jupyter notebook lets you do is that it maintains the memory of variables, for example, string variables, the list variables that you define, and it keeps that in memory, and that memory remains as long as you're computing these cells. So I define a variable a which has Jupyter in it, and I go on to cell number 3, and as you can see, cell 3 has a memory of what was done in cell 2. So as you go along down in a Jupyter notebook, what it allows you to do is, number one, it lets you dynamically compute your resources and dynamically compute your programs. And second, it maintains the memory of what you have done already. So you will not have to compute certain pieces of your code again and again. For example, if you're reading a very huge file, for example, a file which is 2 gigabytes large, that file will not have to be read again and again if you're doing multiple operations on it. Now let's look at the one last functionality before we jump on to VCF files. So you can also display or interact with your bash command line in the Jupyter notebook. So for example, I'm listing the files in my directory, and if I put an exclamation sign here, it lets me perform bash commands, for example, ls. So that was a brief introduction of Jupyter notebooks, and now we will switch back to our demo number 1, which is genomic characterization and data formats. So as Professor Jordan talked about how we have genomic data coming in to pretty much revolutionize the health industry and public health, one of the key things to focus on is how to store this genetic data. So one of the most common type of genetic data that you will see is genotyping data, which essentially has all the variants that exists in a particular individual's genome. And it only holds the variants that we want to look at. So it does not hold all the variants, but it holds the variants that the sequencing technology was looking for. And this variant data is stored in something called a VCF file, a variant calling format file. So we start our demo session with a file called allcombined.vcf. This file has individuals from 1000 Genomes project. This 1000 Genome project is more than a decade old, and it started with sequencing of individuals in different geographic locations, and which represent different populations all across the world. So in our particular allcombined.vcf, we have individuals from 4 different population groups. The first one is the Colombian population group in Colombia. It's represented by CLM. The second one is Iberian population, which comes from Spain. So essentially it captures the European populations. The third one is Peruvian population, and it comes from Lima, Peru. And it again, the idea of the 1000 Genome Project was to capture the Native American individuals living in that part of South America. And finally, we have the Yoruba population from the present country of Nigeria, and it represents the African individuals and their genomes. So we move on to look at what, again, going back to what a VCF file is and what it looks like and what it holds. So we have a command here, which is a fairly simple Linux command, and what it's doing is displaying certain part of a VCF file here in front of you. So what you see here in the beginning is this, all the lines that starts with double pound symbol. These symbols they represent a meta information. The information is additional to our genomic data, and it tells us what all resides in our VCF file. And after that, there's a dynamic part of our VCF file, which essentially holds the genotype data of the individual. So again, for this VCF file, we have more than 200 individuals, and they're all coming from these 4 population groups. And I'm only showing you 4 individuals here if you look carefully. So 8G01112 is an individual code. Again, we cannot show individual name or participant names. So these are represented by codes, and these participants, we have more than 200 of them in our VCF file, but for clarity, I'm only showing 4. And let's jump into the description of a VCF file, and then we'll keep on referencing this part of the program again. So a VCF file, in addition to the double pound comments I talked about, has a dynamic part in the latter half, and it's essentially a file that you can open in Microsoft Excel. And this file has multiple columns, and the first 9 columns are static in nature. These columns will always hold a particular type of information. So let's go over these columns. So the first column is a chromosome number. So it just tells you the chromosome number it is in. The next one gives you the position on that chromosome. So you can see this first entry. This is a position of 160,575 on chromosome number 22, and the third column gives you a variant identifier, any RSID. So RSIDs are coming from dbSNP IDs, and they recognize or they map to a particular variant which is found across a certain human population or across humans. And then we have reference alleles and alternative alleles. The reference alleles tells you what allele is was found on the reference genome, and alternative is what so this particular individual might have. And then you have certain columns for quality scores, for filters. These are again they have to do with how the genotyping was done and what was the confidence of this particular variant. And finally, we have some miscellaneous information where it tells you if it's a single nucleotide variant, or if it is an indel or a deletion. And finally, we look at our starting column number 10, where we see the individual data which is stored. So in the beginning, you saw was the particular SNP and what it was, and now you see if these particular individuals are homozygous or heterozygous for reference allele or alternative allele. So here you see 0/0, and with what it essentially means is this particular individual has a reference allele AA on both sides of their chromosome. So they get their chromosome from their parent 1 and parent 2, and both of those chromosomes have A at this particular position. Similarly, 0/0 again and again. And if you see 1/0, that means one of the parent has a reference allele G and the other parent has an alternative allele T. And if you see 1/1, it means both of them are alternative allele. So we will now go, since now we know that what a VCF file is, we will look into what a PED file is. So PED file is a plain pedigree and genotype file format. This file format is especially useful for multiple tools which analyze the ancestry of a given sample or given individuals. So we convert our VCF files to PED files, and it's a fairly straightforward command using this particular software called Plink. So how to install Plink, the way to install Plink is given in our GitHub, and you can simply install the tool, and you give just one command in which you mention VCF, and you recode it to a PED file. And Plink quickly creates the PED file for you, and a PED file is always accompanied by a map file. And again, if you look at the PED file, this is what a PED file looks like. And it stores information of particular individual and information for their phenotypes and their parents' IDs. And finally, it has the genotype information. And it has its own way of encoding the data. Initially, we saw that the VCF files encode 00. Well, PED file, on the other hand, it encodes 0, 1, or 2. And all these, if you're doing bioinformatics, you will come across multiple different types of file formats, and they all have their way of representing the data. And just another note about PED files, they're always accompanied by a map file, and this map file just tells you what variant information is to be found at a particular position. So first variant will be situated here, and then and so on goes towards the right horizontally. Now, finally, on our last step, we will look at something called a bed file. So bed files, again, are a representation of genotyping data that we have been seeing throughout this demo. But bed files, how they make our life easy is they store the data in binary format. And as you can expect, a binarization of data will reduce the size as well as computational time for any type of analysis we want to do later on. So again, converting a PED file to a bed file is very straightforward using the software Plink. And we just give the command, we tell it to make bed, and it creates the bed file for us. And just a quick note about a bed file, like a PED file was accompanied by a map file, bed file is accompanied by a FAM file and a BIM file. And essentially, if you look at this, all these formats are trying to store information of genotyping of individuals. So these AG01500, it's an individual. We want to store, we have multiple genotyping calls from multiple individuals, and we want to store it as concisely as possible so that it takes less space and it occupies and it saves us computational time later on. And in this notebook, you also have the link to go to the Plink file format descriptions to see more what they mean and what is their importance. And finally, I won't go into much depth into this section, but as I was saying, the VCF or genotyping files are very large in size. So sometimes what we do is called a pruning stage. And essentially what, I'm not going to take much time into this because it's totally another topic. But what it does is it reduces the size of a genotype file depending upon what information is relative. So SNPs are in linkage through whenever in any individual, and we don't want SNPs that do not give us information. I won't go into much detail into this, and you can read more about it. I have some references there, but this completes our demo number 1, our notebook 1. And again, this is just an overview of how the genotyping data is stored. And you can go on our GitHub to see more about it, and there are multiple references here, but this was just an overview of how we store the genotyping data and how we can compress that data, or how we can store that data even smartly to save space and computational time later on. So I will end this demo now, and I will stop sharing my screen. Professor Jordan, if you can go ahead.
Thank you, Shivam. Right on time. Very nice. So we're going to go now to the second session, which is, Shivam, can you see that okay?
Yes, I can. Thank you.
Good. Genetic ancestry. We're going to have 2 sessions on genetic ancestry, and the first one is on principal components analysis. Again, the idea with this presentation is just to give you a brief, high-level introduction to the concept. I'm not going to go into detail. We'll leave you with these slides, which include resources where you can look for more information if you want to learn more. Shivam will go through the exercise on the Jupyter Notebook. And again, you will have the Jupyter Notebook file that you can open. You'll have access to the data that's needed to run the Jupyter Notebook, as well as information on all of the dependencies. In other words, all of the programs that you will need to run principal components analysis. Okay, so let's talk a little bit about human evolution. Humans originated in Africa about 250,000 to 300,000 years ago. Starting around 100,000, 75,000 years ago, humans migrated out of Africa. Modern humans, Homo sapiens, migrated out of Africa and colonized virtually every part of the world. In so doing, they formed new populations. These populations were physically isolated. Because they were physically isolated, they were also reproductively isolated. And so the populations accumulated genetic diversity over time. So if you think about the entire spectrum of human diversity that you can observe with your eyes for populations around the world, all of that is a result of this process, this long, slow march of migration around the world, occupation of different parts of the world, physical isolation, followed by genetic diversification. And that took place for about 99% of the time since humans migrated out of Africa. Okay, so by diversification, I mean accumulation of genetic differences between populations. Okay, now starting around 500 years ago in this part of the world, and by this part of the world, I mean the Americas, and Shivam spoke briefly about this last time, this process of human evolution of migration followed by isolation and genetic divergence was flipped completely upside down. So owing to Columbus' voyage to the New World, followed shortly thereafter by the beginning of the transatlantic slave trade, you had a convergence of European, Native American, and African populations that had been evolving separately for tens of thousands of years. And these populations began to mix and exchange genes. So genetic admixture happens when previously isolated populations come together and begin to exchange genes. Now, of course, we're in the modern era, and technology has greatly reduced barriers to global travel. So humans move around the world with relative ease, and this yields the kinds of cosmopolitan populations that we all live in, which are made up of a mix of individuals from populations that were historically isolated. So this process breaks down reproductive barriers and creates admixture, okay? So what we want to do when we analyze an admixed population like Colombia, a population that has individuals that have genetic ancestry from multiple continental population sources, is have good reference populations. So reference populations are global populations. They're indigenous populations, so they're indigenous to different parts of the world, and they represent the putative ancestral source populations that came together to create the modern Colombian population or the modern admixed population for any part of the New World. So these are populations, Native American populations from the Americas, European populations, and African populations. So we need good reference populations, and we can relate those reference populations and the modern admixed Colombian populations using principal components analysis. So I showed this in my talk today. I'm not going to belabor this, but every point is an individual. They're color-coded with respect to different population origins. And in terms of the Colombian population, the three poles of human genetic diversity are Native American, European, and African. And we see different Colombian populations in green from Antioquia and purple from Chocó, and they show different patterns of admixture. That is, different patterns of combinations of genetic ancestry from these different ancestral sources, okay? In order to do a principal component analysis, we need genotype data. And what principal component analysis does, it takes this very high dimensional genomic data. So by high dimensional, I mean for every individual, you have thousands or millions of variants. So you have a million potentially dimensional matrix of data, and PCA transforms that data to create a new set of orthogonal coordinates, each of which maximizes the amount of variation in the data. So again, I'm not going to go into details, but you can imagine it like this. If these are the raw data, the first PCA maximizes the variation in the raw data. So here we're only looking at two-dimensional data, and the second one is orthogonal to that original axis. So without going into details, again, the big point of principal components analysis is to take this very, very, very high dimensional genetic or genomic data and reduce the dimensionality in such a way that it helps us to visually understand the relationships between the genetic relationships between the individuals in our data set. Okay, so I will leave it to you to look at that. And so here we see three-dimensional PCA, two-dimensional PCA, and I will leave it to Shivam to go through the matrix of genotypes that is used to do the PCA analysis. So we already looked at that last time, okay? And here's a nice example from a paper published some years ago in Nature, Genes Mirror Geography within Europe. Here's a map of Europe with each color color-coded, and then individuals sampled from those countries are color-coded in the same way. And this is a two-dimensional principal components analysis of genome sequences taken from these people. And you can really see the extent to which the genomic relationships among these individuals mirror the geography, again, reflecting that historical pattern of migration and reproductive isolation. It's a very interesting paper if you want to look and understand more about how PCA is used. So here are some additional resources. And with that, I will stop sharing and turn it over to Shivam for the practical demonstration of how it is that you do PCA analysis. Shivam?
Right. Thanks again, Professor Jordan. And I'm just briefly going over what Professor Jordan just mentioned. The idea of PCA is to reduce the dimensionality of our genomic data. We already saw that this genomic data is very, very large in size. even though it captures just 1% or less than 1% of our overall DNA, but still it's very large in size. And the idea is to reduce the dimensionality of this data so that we can visualize it more properly and see different patterns. For example, like Professor Jordan showed, that we can see pretty much a geographical map of Europe using this genomic data. So in this demo on this notebook, what we'll be going through is converting our existing VCF data. So if you remember from demo number 1, we had a VCF file from which had individuals from 4 different populations: Colombia, Peru, Yoruba from Nigeria in Africa, and Spanish population from Europe. We have these 4 populations, and the idea is to see how these populations look like on a PCA map, how far these populations are from each other when it comes to genomic data. So we'll start with where we left in our last demo. So we have a VCF file which we converted into a bed file. The bed file is again a binary representation of the data, and it's accompanied by a BIM and a FAM file. So you have the commands in demo number 1, and you can go and convert the VCF file into bed file and the FAM and BIM file. And these are the files which we will be using for this notebook. So once we have the bed files for our genomic data, what we do is, again, we go back to our friend Plink, and we perform PCA on it. And Plink again gives us a very simple functionality in which we give it the bed file, and we can run the PCA on it, and it produces a nice little output of eigenvectors and eigenvalues. So the way PCA is performed is it tries to find the axes, the orthogonal axes, which are obviously at 90 degrees, but which explain the data in the best possible way. And then it scales it onto n number of dimensions. So at the end, it produces something called eigenvectors and eigenvalues. And these hold the information coming from the PCA. So once you have these two files, what we will do, at least in this notebook, is we'll jump onto our programming language. So if you are in a Jupyter notebook, you can change the kernel here using this button. So you go here, you can select whatever kernel you want, Python 3, R, and you can set it up. And now the cells in your notebook will be executed using that particular language. So in here, in step number, we go to step number 2 of our notebook for creating PCA plots. So we import the necessary libraries, and this is just how it shows that the libraries are imported. And again, these cells of the notebooks that you will see on your screen now, moving forward, these are computed real time for you at our server. So I'm doing this computation simultaneously as I'm presenting. And this is the beauty of Jupyter Notebooks. We don't have to save the data. Everything is dynamic and it's running on the server in the background. So we start with importing our libraries here. And after that, what we're doing is that we import the eigenvectors. So if you remember, I told you when we run Plink on PCA, when we run Plink on our bed files, and we ask it to perform PCA, it gives us eigenvectors and eigenvalues. So first, we'll import eigenvectors into our data frame. And what we will do is we'll modify the data frame a little bit so that it suits our requirements. Essentially, we will be able to read it better we will be able to read the data frames better this way. So moving on, what we'll do here is let's have a look at the eigenvectors here. So eigenvectors I'm only showing the eigenvectors from PC1 and PC2, but originally there are 20 eigenvectors present for all PC1, PC2, PC3, 4, 5, all the way till PC20. So PC stands for principal component. So principal component 1 and 2. These are the ones we will be looking at because these are the ones that are in fact the most important. And I'll show you why they're the most important. But this is what our eigenvector looks like after we have done a little data manipulation and data cleaning here. So it has individuals and their IDs, and it has PC1 values and PC2 values, and we have it for all the individuals. Remember, we have individuals from 4 different populations: Colombia, Nigeria, Spain, and Peru. We have 4 different populations in our VCF file, and we have, as you can see, we have over 300, in fact, almost 400 individuals here. Next, what we will do is we will get the population group of this data. So originally, if you remember, we know that what these individuals are, which population these individuals are coming from. So we know that, for example, this HG01501, this individual is coming from population group IBS. We know that IBS stands for Spain. We know that this population group, this is what these come from. So we're going to annotate that information, and what will it allow us to do is it will allow us to see what population groups, what the ancestry looks like for different population groups. So in this particular cell, we are just reading the files, and we're merging them, we're joining the file back to our data frame, and now we have population group data. Next, what we are going to do is we are going to plot a PCA plot, and it's essentially just plotting the 2 dimensions, PC1 and PC2. We have 2 columns here, and what we're doing is we're just making a scatter plot to see what the populations look like. And we have encoded the colors for each population in this particular R code. And once we run this code, what we get is we get a nice little PCA plot, and this is pretty similar to what Professor Jordan had showed in his slides. So we see that YRI population, which is represented by blue, and remember YRI is from represents the current population living in Nigeria, and they come across in this little cluster here on the PCA. And then we see, if you see behind these green dots, there's orange dots, which represents the European population in our data sample. And then finally, we see the population of Peru, which represents the Native American ancestry here. And the final population is the population from Colombia. And as we can see, it's a kind of a mix between Native American and European ancestry. So these individuals have ancestries of both European and Native American. And some of them also have a significant ancestry from the African group. So as this green dot goes closer and closer to blue, it says that the African ancestry is increasing, it's reaching to that cluster. So this is a nice way to represent or see what our population looks like, or the population of our data looks like. And after that, what we can do is we can use our eigenvalues. So PC1 and PC2, the way the algorithm works is that it focuses on reducing the dimensionality, and tries to find very few number of dimensions, maybe 20, maybe 2, maybe 3, so that you can explain the data as good as possible. Again, we cannot have so much genomic data, and again, we cannot visualize it. The idea of PCA is to compress the data and make it visualize and make it in a way and transform it into a way that we can visualize the data. So again, we don't, but now the question comes, is PC1 and PC2 good enough? As I mentioned, Blink gives us 20 PCs initially. So are PC1 and PC2 good enough? Or do we need PC3? Do we need all the way till PC10? Like, how do we know how many PCs are, or what PC1 and PC2 are able to explain? How good are they? So for that, we use something called eigenvalues. Again, remember, Blink produced 2 sets of files for us. One was eigenvalue and the other one was eigenvector. We used eigenvector to produce our PCA plot. And now we will use eigenvalues to see how good do these PC1 and PC2 are working. So we import these eigenvalues into our database and into our RStudio data frame. And then we plot something called a scree plot. Scree plot is another type of a graph which tells us how much the variation in the data is explained by respective principal components. So if you see here closely, the first bar represents PC1, then PC2, 3, 4, 5, and onwards. So we can see in our scree plot, which is again coming from eigenvalues, that the first PC is able to explain much more than PC number 3, 4, or 5. And PC1 and PC2 are probably able to explain the most of the variation in the data when compared to other PCs. And that is primarily the reason why and along with that, we are not able to visualize, obviously, data in multiple dimensions. So if we can possibly do a PCA in a 3D PCA plot, but after that, we cannot visualize it. So it tells us that how important or how much variation our PCs are able to explain in the data. So that brings us to the end of demo number 2. I know it has ended a little earlier, but let me stop sharing the screen.
Good. Thank you, Shivam. And it's perfectly fine that we're on time. Augusto, we're doing good with time. I don't know if it's possible to take any questions. And I know you just logged in shortly ago, but I don't know if
Let me check, review if there are questions from the audience.
Okay.
Just hold on a second. So far, there is none, but it's so far there is none, but perhaps what we can do is to, ask the participants to share the questions in the chats so we can see them and we can try to answer them. This is a good time to do questions. Come on. Try to do it. Okay, it seems we don't have any questions, so we can move ahead.
Good. Okay. Thank you, Augusto, and thank you, Shivam, for that. So we're going to stick with the theme of genetic ancestry, and we're going to move on to admixture analysis. So I'm going to share my screen. Good. I hope everyone can see that okay. So I talked about admixture before, and now we're going to go into more detail, okay? So we're still talking about how it is that we can infer ancestry at the genome-wide level. So again, what we're talking about here is comparing our participant genome sequences of interest, so the Colombian population, against putative ancestral source populations, global reference populations that represent the putative ancestors of Colombians. And given what we know about the history of the conquest and colonization of the Americas, we can infer with confidence that most of the ancestors, not all, but most of the ancestors of Colombians come from Africa, from Europe, and from the Americas, okay? So we approximated that with principal components analysis, PCA, but PCA doesn't actually quantify the ancestry of the individuals. In other words, it doesn't tell us what percent African ancestry does someone have, what percent European, what percent indigenous or Native American. What PCA does is it reduces the dimensionality so that it's easy to manage and we can visualize the relationships. And when we visualize those relationships, we saw evidence of admixture, that is, individuals that grouped between the three poles of human genetic diversity, African, European, and American, okay? But if we want to know what are their fractions of ancestry from each of these groups, we need to do admixture analysis. And on this slide here, you see a typical admixture plot. This is straight from the talk that I gave this morning. You have African populations in blue, European in orange, and then indigenous shown in red. So these are the reference source populations from Africa, from Europe, and from the Americas. And then we have modern cosmopolitan populations from Medellin, from Choco, and an African American population from the US. And remember that each one of these bars, each one of these columns is a single individual, and the relative size of the different colors correspond to the percent or the fraction of ancestry from each of those groups, okay? So admixture, that's the name of the program in all caps, is an unsupervised classification technique that uses observed genotypes. So it doesn't need to know what the ancestral populations are a priori. Okay, so what admixture wants as input is a matrix of observed genotypes and then a value for K. And Shivam is going to talk more about this, but K is just how many different theoretical ancestral source populations are you going to infer. And you can use a priori knowledge to define K. In other words, you can say we expect a priori Colombians to have 3 ancestral source populations. So we'll use the value of 3. Or you can run an admixture across a range of K values and look at which value of K yields the best fit to the data, okay? And what admixture does is it finds 2 things from these inputs. K, in the case of Colombia, 3 sets of allele frequencies that are the allele frequencies from the ancestral populations, and the fractions of each of the K sets of allele frequencies within each individual. And that represents the admixture ratio for each individual. So this is the formal mathematics of how the algorithm works. So what it does is it optimizes those allele frequencies and those fractions to give the most likely probability for the observed data, okay? And then it turns out, and Shivam will show this in the exercise, that it's up to the user to figure out what the inferred populations and what the inferred fractions mean. And by that, what I mean is, remember that admixture does not need to know the ancestral populations a priori. So unlike other genetic ancestry inferences techniques, you're not assigning a group as an African reference population. You're including African reference populations and European and Native American, but you're not telling the algorithm which is which, and it's going to partition the allele frequencies into 3 groups and assign each individual a fractional assignment for each one of those 3 groups. And then based on what the data show, you decide which one of those corresponds to African ancestry, European, or Native American in the case of Colombian ancestry, or other combinations depending on what you're looking at, okay? So this is just an example of what you would get, what admixture outputs for a few individuals, right? So we have a Yoruba individual. This is an African population. We have Zapotec, which is a Native American population. We have British, and then we have 2 admixed individuals from Chocó and from Medellin, okay? So if you look at this, you can begin to guess which one of these 3 ancestry components, F1, F2, or F3, corresponds to the ancestries that we're interested in, right? So this individual from Africa has almost all of their ancestry from F1. So you can infer with confidence, especially if you look at lots of individuals, that this is the African component. Likewise, this indigenous individual, and I'm only showing one, but we would do this for lots of people, has almost entirely the F3 ancestry component. In other words, the allele frequencies for this individual correspond best to this ancestry component. And the British has mostly F2. And then you can look at the admixed individuals from Chocó and Medellin and infer their patterns of admixture with respect to these 3 ancestry components mapped on a posteriori, after the analysis is done, onto the ancestral reference populations, okay? So we figured out that these are European, African rather, European, and Native American, based on the relative fractions of ancestry for the reference individuals. I hope that's quite clear. And finally, here are some additional resources. So with that, again, all of these slides will be available to you, and I will turn it over to Shivam to show you how this is actually done in practice. Shivam?
All right. Can you see my screen?
Yes.
Okay, perfect. Thank you so much, Professor Jordan. And I'm just going to, as Professor Jordan mentioned, I'm going to give a quick demo of how those ancestry fractions were obtained and how can we produce those ancestry plots, admixture plots, which he showed on his first slide of his presentation. So,
Shivam,
yes.
I'm sorry to interrupt you, but we need to make profits. There is a one question already from the audience, and I think this question is right from King Jordan. Talk. So to you two, King and Shivam, somebody's asking here. Let me just say the name. Jorge Mario Vasquez is asking how much computing capacity is required for doing an analysis like this one, this kind of analysis we're doing today.
Yeah, that's a good question, and that's actually, it depends on several factors, and it's actually more of a question for Shivam since he does the analysis. But it depends on how dense your genotype panels are, and what does that mean? That means how many individual variants are characterized for each individual, and how many individuals you have. So let me return to number 1 first, because this relates to something that Shivam talked about right at the end of the first session. So one of the things that you want to do always when you do these kinds of ancestry inference analysis is what we call linkage disequilibrium pruning. Okay, now we're not going into that in detail, but what that means is the following. So this gives me a chance to elaborate a little bit on that. For any region of the genome, any haplotype, you have a number of different variants in that chromosomal region, but their allele frequencies will be correlated across individuals and maybe even across populations, because all of the variants in any given segment tend to be inherited together. Okay, so what that means is if you have one variant in that region, or 2 or 3 or 5 or 10 or 100, you don't necessarily get more information, right? Because they're all linked. They contain mutual information, so that you only need one or a few variants in that region. So what linkage disequilibrium pruning does is it reads along the genome, and then it chooses a single variant for each window of the genome that you define that contains the maximum amount of information for that region. So the reason why I'm explaining that is because in order for Shivam to answer your question, I think we need a kind of prologue to say that it depends on how many variants and it depends on how many individuals. Now, depending on what you want to do, you probably can't control the number of individuals you have, but you can reduce the number of variants by doing linkage disequilibrium pruning. You want to do that before you do PCA. You want to do that before you do admixture analysis. And in the Jupyter notebook provided by Shivam are instructions for how to do so. Now, I'm also going to give a high-level answer before I turn it over to Shivam. So these methods for ancestry inference, well, maybe not so much PCA, but admixture work very, very well, but they don't scale well to biobank-sized data sets. So if you have many thousands of individuals, it is slow and it takes a lot of compute capacity. But the algorithm admixture was actually made to be an improvement on a previous algorithm structure, and it actually works a lot quicker and a lot better. So I know that's kind of a high-level answer, but Shivam, can you now step in and give a little bit more detail on what level of resources they need to do this kind of analysis?
Yes, absolutely. All right. I'll just quickly share my screen to give a bit of context while I'm talking. And again, it's a good question. And as somebody or as a bioinformatician, it is important for us to understand how much time will it take to perform a certain task. So as Professor Jordan said, when it comes to getting the ancestry fraction of n number of individuals, one of the primary things that depends on which affects the time is, of course, the number of samples. So right now we have around 400 individuals. If you go to something like UK Biobank, you have 500,000 individuals. That's almost 1,000 times more. So obviously, the time will keep on scaling, and sometimes it scales exponentially as the number of samples increase. The other thing to note again is the number of SNPs or number of variants that we are using. So for example, this particular algorithm, which is goes by the name of admixture, it's a fairly, I would say in my experience, it's one of the faster tools to do it. And it gives you the overall ancestry fraction of the individuals you give in. This algorithm works well with just 10,000 SNPs or 10,000 variants. So this is what the author claims, that you can just supply 10,000 variants, and this algorithm will be able to, distinguish and classify different ancestry groups in each individual or each sample that you supply. For me, for this particular calculation, admixture took around 2 to 3 minutes, and I was again using I think somewhat a little bit under 15,000 SNPs. I performed linkage LD pruning using Blink, and admixture ran fairly fast for 400 samples. I did not use any multi-threading, although admixture does give us an option for multi-threading, but it ran under 2 to 3 minutes as far as I remember. And again, but
Shivam,
yes,
this is King. Can you tell us what kind of machine was that? How much resources did you have? And well, tell us that first, and then I have another comment.
Sure. So the machine we had was, in fact, a really good one. The machine had almost 500 gigabytes of RAM and 24 processors. But our process, it did not use more than one core when I was computing it. So you can supply the number of cores to use using this particular argument. But the way I ran it, we did not use any other core. But the way I was able to bring down the computational time was by using just around 15,000 SNPs. Originally, the number of SNPs were far higher. They were, I had more than 2 million SNPs. So yeah, Professor Jordan, if you
right. So I was going to say that the way we have set up these exercises, so remember that we're providing everything for you on this GitHub resource. So that includes the talks that I'm giving, you know, the short slide deck with additional resources, the Jupyter notebook that Shivam is going through, but also the data. So we recognize the computational limitations. So what we did is we chose the data in a very intentional way to make the data sets manageable in size. So if I recall correctly, Shivam, for the admixture analysis, you only used a single chromosome and you used a small chromosome, right?
Exactly. I used the smallest chromosome, chromosome number 22, and I pruned that chromosome, in fact, and brought it down to 15,000. So we really brought the data down, and that's why it's able to run in 2 to 3 minutes.
Right.
Otherwise,
I just say that so that the students are aware that these are probably artificially small data sets so that you can manage them and test them out. And so it does run pretty fast, but as you get into bigger data sets, you will need robust computational resources to run these kind of algorithms. What we've tried to do is set it up to make it as easy as possible for you to run it. So could this be something that could be run on a laptop, like with OS 10 if they have a Mac, or if they have Linux installed on their laptop?
Yes, I believe so. So a fairly new laptop should be able to handle this particular data set that we have, because we really pruned it down just for, you know, demonstration purposes.
Good. Any other questions, Augusto?
No, not so far.
Okay, good. I mean, the more interactive this can be, the better. So I like the questions. So if we can encourage who's ever logged in to keep going with questions, I much prefer to have good questions than to race through these exercises. So with that, Shivam, I'll turn it back over to you.
All right. Thank you, Professor Jordan. And I'll go back to the start of demo number 3, which is using the admixture tool to get the ancestry fractions and further create some admixture plots. So we begin this particular algorithm, or we begin this notebook with running admixture. So running admixture is fairly straightforward. It's not complicated. All you have to do is just one command. The thing that the thing that makes it more complicated is giving it the right number of K. So K, as you can see here, is the number of theoretical source populations. So Professor Jordan talked about this in his presentation, where he described how Colombian population we propose, or like we have reason to believe that Colombian population has 3 theoretical source of populations. Number 1 is the Native American population, which is number 2, it's the European population, and number 3 is the African population. And we have groups representing all these populations. So that's why when we ran admixture, we gave the K as 3, the number 3, because we believe that there should be 3 clusters of populations. I would like to remind everybody that all this data, and Professor Jordan mentioned that too, this is all the results I have, what I'm demonstrating right now. All of that has been executed on chromosome 22, and it has been pruned down to mere 15,000 SNPs. That is the lower limit of admixture. Admixture is able to perform well for at least 10,000 SNPs. So this is for demonstration purpose. And this command shows you how we run admixture. And moving on, when admixture completes its run, it produces 2 files. And the file names are whatever output name you give, .q and .p. And .q is one of the files of interest, at least for our demonstration or our notebook right now. .q file, it contains the ancestry fractions. And if you want to go in depth, you can also look at the p file, which has the allele frequencies or the painting vectors to see what they look like. So admixture supplies us with this q file, and we immediately go to R programming. So just a quick reminder, you can go here on Jupyter Notebook and select your kernel. For this notebook, you're using R programming language. And we start by importing libraries. So these are standard libraries which people use, and we import these libraries. Moving on, we import the individuals to population data set. So we import that data set into our data frames to save them into variables. And I will show you what each data frame looks like. So we capture the individual to population group data. It says in the variable right here. So what this has is it has individual IDs and it has the population group. So for example, this particular individual has population group IBS annotated against it. So what it essentially means is that, this individual the individual sample was obtained from Spain. So it probably has a lot of European ancestry. And similarly, if you look at the other end of our data frame, you can see now we have Colombian, and this data frame is, again, it has 400 samples, and I'm just showing a little part of it. I'm just doing the tail on it. And this shows us that, these individuals are annotated as Colombian population. So we have 4 populations coming from Peru, coming from Colombia, and Yoruba or Nigeria in Africa, and Spain in Europe. Next, we look at the FAM data. So one of the things about admixture is, and I would like to highlight that, is admixture does not give us, it simply gives us ancestry fractions. It does not tell us what these ancestry fractions are assigned to which individual. So we have to see what data frame we gave to admixture. So we gave a bed file here, and we have to annotate accordingly, like what ancestry fraction belongs to which individuals. We supplied 393 individuals. We need to know what ancestry fraction comes from and for which individual it corresponds to. So all this I'm doing here is trying to just merge the data. And now we have joined this data and we have the exact order of the individuals. And now we bring in the admixture output file. If you remember the Q file here. And the Q file, it represents 3 here, which is the number of K value we give. K is the number of theoretical populations that are believed to be in a population group for which we are running admixture. So we look at this particular data file, and as you can see here, we have the ancestry estimates here. So I can quickly show you how these ancestry estimates look for different individuals. So if you can see, we have ancestry estimates for all the 394 individuals we had. And again, these are coming straight from admixture, but as you can see, it does not tell us, for example, this particular row, which individual does it correspond to? And that is essentially what we're trying to do here. So in the next step, what we'll do is we'll combine everything together. So a little bit of error here, but I'll quickly move on to the next thing. So we combine the data there, and what we do is, if you look closely, you will observe that admixture also does not give us the label for K population cluster it creates. So when we saw these populations, if you look closely, it does not give us the labels here, V1, V2, V3. Now, we do not know what V1 is or what V2 is. Is it European? Is it African? Is it Native American? So, and this is precisely why we use 3 reference populations. Again, IBS, PEL, and YRI. And we have we have reason to believe that these are the 3 source population for Colombian individuals. So now what we will do is we will try to merge this data with the ancestry data we have, and so we summarize this data on a data frame here, and we'd like to see what V1, V2, and V3 look like for different individuals. So the data is annotated now, and as you can see, CLM, we're not sure what V1, what percentage it will be inheriting from V1, V2, or V3, but we do know IBS, it's 99% V3. And similarly, YRI is 99% V2. So we have reason to believe that V3 is in fact European, which is coming from the Spanish individuals in our cohort, and V2 is coming from African, which comes from the Yoruba populations of Nigeria. And similarly, we assign V1, which is 87% Peruvian. We assign V1 to that, and that is coming, we assign Native American to V1. And finally, now what we have here is we have assigned the labels successfully here. So this is where it pretty much brings us to a more understandable data. What it means is we have cluster labels for each data set here: Native American, African, European. So let's try to see if we can look at the Colombian population here. So just give me a quick second. All right, so this is was able to fix the error, and this is again what we went over. Now we will group them again, and we have the average ancestries for each individual. And now we will have a look at the Colombian population. So Colombian population individuals. So here we are. So this particular cell represents all the Colombian populations here and their ancestry estimate. I would like to remind that here are the labels for V1, V2, V3 in column 1, column 2, column 3. So you can see, for example, YRI individuals are all of them are 99.99% African, which is what we expect. That is why we sent them to admixture tool. Next, we look at the Colombian individuals, and we see there's a fair mix of them between the three ancestry estimates. And that is in fact what we saw in one of the slides of which Professor Jordan shared with us in his presentation. So the individuals from Colombia and they have varying ancestry fractions from all three populations, and we're able to see that. Now what we're going to do is we're going to again just to see what average percentage do Colombian individuals look like. So it's 66% European, 6 to 7% African, and the remaining 28% Native American. And again, one thing to keep in mind here is that all these individuals, the data we used was significantly LD pruned. We brought it down to 15,000 SNPs, and there was only one chromosome used, which was chromosome 22. So if you look at these data, there won't be a very precise match to the gold standard out there, but it captures but for demonstration purposes, we brought down the data, but it captures the overall picture of what the ancestry fractions would actually look like. Now, once we have the ancestry fractions, what we are interested in doing is we want the pretty admixture plots that Professor Jordan shared with us in the beginning. So we just decompose the matrix using a melt function from R, and what it does is it decomposes the individual into each row, and moving on, we determine the colors for individuals, and then we use ggplot from R to produce those admixture plots. And as you can see, again, this is running on our server in the background. I just ran it again. I can run it again. And you can see here, the orange denotes European, blue denotes African, and red denotes Native American. So the IBS and YRI, which is IBS from Spain and YRI is from Nigeria, they both represent almost 100% European, and for YRI, it looks almost 100% African, except for one little individual there. And for Peru, it looks like a fair mix between European and Native American ancestry. It captures Native American ancestry for the most part. And here is the admixture plot for the Colombian population. So you can see that there's a few individuals who have significantly higher Native American, and then there are a few individuals who have significantly higher European population, and it's the K that we supplied according to that. The ancestry fractions are deriving from 3 separate clusters: African, European, and Native American. So that brings us to the end of this notebook. One essential thing here is you might want to write this data. So this data is, which you see here, is ancestry fraction. It's all annotated according to the individual ID and according to the population group. So you might want to write it. If you're running this notebook, you might want to write it on your system to have that data for you. And so before I conclude this, just to recap everything that we went over, we started with the admixture tool. We give it the number of K that we think, or we have a theoretical reason and a confidence to give that particular K value. So we were looking at Colombian individuals particularly, and we have scientific reason to believe that they derive from 3 theoretical population groups or clusters, you can say, and they are Native American, European, and African. And we run an admixture on it. It gives us a file. The file needs some data manipulation, which we do. We see what average ancestries look like for different individuals. And this is an important step while running admixture. You have to see and you have to assign the cluster label. Admixture does not give you the cluster label. It says V1, V2, V3. That's not very helpful for us. What we have to manually do is assign the variables for each of these clusters. So we put Native American for V1, and we saw Peruvian individuals were having 87% of V1. So we had reason to believe that this particular cluster is Native American. And V2 and V3 similarly, they showed 99 again percentage of ancestry for these clusters. So that's how we annotated the clusters. We combine the data. There's some data manipulation happening. We melt the data set, and finally here's a ggplot function which sort of brings you back the admixture plots. And that concludes our module number 3. I would welcome any questions, and Professor Jordan, if you have any questions or any additional remarks to make here.
I don't, Shivam. That was very clear, but I will turn it over to Augusto briefly to see if we have any questions, Augusto.
So far we don't. Let me see in the Excel, please. Hold on a second. No, we don't. Actually, we don't have any questions.
Okay,
from the audience.
These next modules are a little longer, and I know we're ahead of time, but I'm anxious to see how much ground we can cover. So I'm just going to jump into the next one. Is that okay, Augusto?
That's very okay. Please go ahead.
Okay, so let me share my screen. Okay, so remember that in order to be able to implement precision public health in Colombia, or really anywhere in the world, we need to have a deep understanding of the relationship between population, genomic diversity, genetic ancestry, and determinants of health. And those determinants of health are individual genetic variants that are associated with either disease or how patients respond to medications. So the first part of this, the first 3 sessions was some basic information on how genetic variants are encoded such that they can be analyzed bioinformatically, and then 2 sessions on genetic ancestry. And now we're going to change gears. The last 3 sessions, we have 2 that are on associations of genetic variants with disease, and then the last one is on pharmacogenomics, associations of genetic variants with how individuals respond to medications. So this first session is on the genomic diagnosis of rare disease, and I'm just going to introduce this and then turn it over to Shashwat. Now, this plot is something quite similar to what Claudia Latig showed in her talk. It's the relationship between penetrance, which is essentially the probability that the presence of a variant will cause disease, and the allele frequency, how rare or common the variant is in the population. We're talking about rare disease, also known as Mendelian disease, sometimes orphan disease. And for these particular genetic variants, they have very high penetrance. So if you have one of these variants, you are very, very likely to have disease, but they're very low in the population. On the other end of the spectrum are variants identified by genome-wide association studies that are associated with complex common diseases. Those have low penetrance, so each variant only contributes a little bit to the risk of disease. And if you have a variant, it doesn't necessarily mean you have disease, but they're found at higher frequency in the population. When we talk about polygenic risk scores, we'll talk about this end of the spectrum, but for here, we're focused squarely on this end of the spectrum of this relationship, okay? Now, let's talk briefly a little bit more about human genetic variation. So all human genetic or genomic variation originally comes from mutation, and mutations are just changes in DNA. So those changes in DNA generate variances, differences in the DNA sequence between individuals. Once the DNA is mutated, once it's changed, if this happens in the germline, those variants can be inherited and spread in the population. And if they rise in frequency in the population above a certain threshold, typically 1 or 5%, they become polymorphisms. So polymorphisms are just genetic variants which have risen to a certain frequency in the population. So remember, we sometimes talk about single nucleotide variants and single nucleotide polymorphisms, SNPs. They're similar, but the difference is just that SNPs exist at a certain frequency in the population, okay? Now, there's lots of different kinds of mutation. I'm not going to get into that, but you can look at this topic. Here's a resource for you if you want to look at that more. And remember, for this demo session, we're just focused on single nucleotide variants. Okay, so what are the consequences of human genetic variation? So when I talk about consequences, I'm referring to what I call first order consequences with respect to the effect of these genetic variations on the proteins that they encode. So these are protein coding sequence variants, and we want to understand how the variations change the proteins that they encode. Okay, now this is a little bit overly simplistic, since non-protein coding variants, that is variants that fall outside of exons, can also impact the function of the genome. So an example is microRNAs or long non-coding RNAs. But for the moment, there's much less known about the phenotypic consequences of non-protein coding variants. So we're focused on single nucleotide variants, even though that's just one class of variation. And we're focused on single nucleotide variants that fall into protein coding regions, even though the vast majority of the genome is outside protein coding regions. Those are just some things for you to be aware of. Now, this is the genetic code. Many of you have likely had this in your molecular biology class. And the only thing I want to do here is just emphasize that the genetic code is redundant. And by redundant, I mean that many different codons can actually encode the same amino acid sequence. Okay, sorry, I had a little interruption there. So the genetic code is redundant. Now, what does that mean with respect to mutation and human variation? There are some mutations, there are some variations or substitutions that are so-called synonymous. And by synonymous, I mean that these are DNA changes that do not change the encoded amino acid. They're also called silent mutations. On the other hand, we have non-synonymous mutations or substitutions. These are changes to the DNA that do change the encoded amino acid, okay? So these include missense mutations that change the identity of the encoded amino acid, but they also include so-called nonsense mutations that introduce a stop codon and or frame shift mutations, which change the reading frame of the open reading frame, which typically leads to a stop codon nearby downstream, okay? So this is an example of different kinds of mutations. So we have a silent mutation, which is a change in DNA, but it encodes the same amino acid because that third position of the codon is redundant. Here we have a nonsense mutation whereby a change to the DNA sequence introduces a stop codon. And here we have missense mutations wherein changes to the DNA sequence change the encoded amino acid. And all of these are going to have, these are the first order consequences of DNA mutations in protein coding regions, and they can have higher order consequences with respect to changes in the sequence and the structure of the proteins that encode these mutations, okay? And we can think about this in terms of what is known as loss and gain of function mutations. Loss of function mutations and gain of function. So missense and nonsense mutations can lead to reduced or lost protein function, okay? So loss of function mutations, these are mutations that essentially knock out the function of the protein, tend to be recessive, so that you need 2 copies of these mutations to get a total loss of function of the protein, because if it was 1 copy, you would still have the other copy to encode the function, okay? Now, loss of function mutations are more common in disease, but they're rare in populations because they're selected against. So computationally, these are predicted as mutations that 1, introduce a premature stop codon, 2, cause a frame shift, or 3, alter essential splice sites. So splice sites are sequences in gene regions that help determine how the pre-messenger RNA is spliced into the final mature messenger RNA. Again, it would entail a whole review of molecular biology to go over that, so I'm assuming you have that information. Now, missense mutation can also infer enhanced activity on proteins. And so by gain of function mutations, we're referring to mutations that elevate the function of the protein. So those kind of mutations tend to be dominant, and they're also more rare in disease and rare in populations. So loss of function mutations are more common in disease, but very, very rare in populations. Gain of function mutations are both rare with respect to disease and rare in terms of populations. Okay, so now that we understand the sources of human genetic variation and what I'm referring to as the first order consequences of human genetic variation, in other words, how is it, what are the different ways that protein coding sequence variants influence the DNA? How can we now drill down to understand the clinical implications of human genetic variation? So that's exactly what we want to do. Imagine you have a laboratory like Juan Esteban Gallo, Genoma Ses. You were given the sequence, the genome sequence, or the exome sequence of a patient that has been diagnosed with a possible genetic disease. You want to understand what is the mutation, or what are the mutations that are most likely to be causing that disease? In other words, how do you know which mutations may cause which diseases? Okay. Now, a caveat here is that since we're dealing with rare and penetrant variants for these monogenic Mendelian diseases, in other words, 1 mutation in 1 gene causes a disease, we are assuming causality here. Okay. And causality is a much stronger relationship between the mutation and the disease than associations, which can be more probabilistic. Okay, so ClinVar is an example of a publicly accessible database. This is widely used, and it stands for clinically relevant variants. So this is one of the first things we want to take into consideration when we're analyzing the genome or the exome sequence of a patient to try to look for mutations that are linked to monogenic Mendelian disease. Lots of these mutations have been characterized in patients and populations around the world, and when physicians characterize these mutations, or clinical geneticists, they populate them in these databases, ClinVar. Now remember, from Juan Esteban Gallo's talk, and also from Claudia Latti, a very, very interesting phenomenon is that a number, I don't know if it's many, but quite a few of the mutations that they're finding in Colombian patients are not found in these databases. And that makes genome diagnosis of disease in Colombian patients more challenging, and has to do with the bias in the genome sequence databases. Anyway, ClinVar works by community submissions to the database with supporting evidence for clinical relevance, and it classifies variants into one of 5 different classes. So benign, likely benign, variants of unknown significance, likely pathogenic, or pathogenic. And so for variants that you want to associate with disease, you want them to be pathogenic or at least likely pathogenic. Now, the ACMG, which stands for the American College of Medical Genetics and Genomics, has set a set of global standards that are used to interpret the sequence variants. And there's lots and lots of information that goes into this. I'm not going to go through this in detail. All of this is here for you as a resource, But as Shivam goes through his demonstration session, you're going to see how these standards are implemented in a specific case example. So there's lots and lots of different levels of evidence that are used to annotate these variants and to interpret these variants in order to make a diagnosis. And at this time, as far as I know, it is simply not possible to automate this. So this takes human intervention. It takes the eyes and the brains of someone that has a lot of experience with this to make clinical interpretations. So all of this is provided for you. I'm not going to go through this, but you can look into this and you can look at the paper if you want more detail. What I am going to do is introduce a case study, and then Shivam is going to go through this case study. So we're going to do this for all three of these sessions: rare diseases, complex common diseases with polygenic risk scores, and pharmacogenetic variants, using the Cuenta de Alto Costo database from Colombia. Okay, so Cuenta de Alto Costo is a non-governmental organization that works very closely with the EPS, the Sistema de Salud, the health system of Colombia, to characterize diseases, prioritize diseases in Colombia that have a maximum impact on public health and the economy. And all of the examples that we're going to go through come straight from the Cuenta de Alto Costo. So these are all examples of diseases that have been prioritized by the Colombian government and this non-governmental organization as having an outsized impact on the economy and the public health system in Colombia. So for this example, we're going to look at hemophilia, okay? So let's imagine we have a case study. So we have a patient and they have symptoms of excessive bleeding, of very large bruises and joint pain, and we suspect we have a preliminary diagnosis of hemophilia, which is a disorder of blood clotting. So the blood doesn't clot normally, and when it can't clot normally, you get excessive bleeding, or after injury or damage, and you can get these deep bruises, blood in the urine or stool, and pain in the joints. Okay, so we have this preliminary diagnosis, but we're going to use genome sequencing, exome sequencing to go in and look and see if this patient does in fact have any mutations that are associated with this disease, or since we're looking at single gene Mendelian disease, any causal mutations that are known to cause the disease. So with that, I will stop and turn it over to Shivam, and he's going to take you to the steps that will need to be done to confirm this diagnosis of hemophilia in a patient for whom we have a whole genome sequence. Shivam?
All right. Yeah. Let me share my screen. Thank you so much, Professor Jordan, for going through those slides. And we're going to go to demo number 4. And this is where we sort of switch our directions into what we have been looking into in the last 3 notebooks. So as Professor Jordan talked about him in his last couple of slides, we have a patient and he has unusual blood clotting in his body or in their body. And the preliminary diagnosis is hemophilia. So, and we have the genotyping done on this individual, and this individual, we have the VCF file which stores the genotype information for this individual. So our agenda or our main goal here is to see what might be the cause of this disease. Could it be a pathogenic variant? Could it be a deleterious mutation? So our goal is to see what might be causing the disease of hemophilia. So we start with this session with a VCF file from our patient. So we have been supplied by their VCF file, and we know that the mutations associated with hemophilia primarily occur on X chromosome, the chromosome number 23. So in order to save computational time and save some space on our computers, we will only analyze the variants that are present on chromosome X of our patient. So the original VCF file that we have, it's like we get it from the individual, we extract chromosome X from it, and we have a VCF file again. So let's have a look at chromosome, the VCF file from this individual. So here you see patient 101 is there. That's one patient. There's no other patient. So this VCF, this is a little different than the VCF files we have been observing in our last 3 notebooks. So in this VCF file, there is only one sample. There's only one individual. It goes by the ID of patient 101. In the previous notebooks, the number of samples were 394, which are coming from 1,000 genomes. 1,000 genomes, again, it's for research purposes. This particular VCF file we are getting from a patient. And let's have a look at their X chromosomes. This VCF file will give us the variants present on X chromosome and the variants that were fetched through genotyping on their X chromosome. So you see chromosome number 23. Sometimes in the VCF file, this 23 will have X here, but right now it has 23 in this one. And it gives you again the homozygous, it gives you if it's reference allele or alternative allele and whether it's homozygous or heterozygous for this particular variant. And again, this VCF file is just the head of it. So we're just seeing the first 10 lines. It's a pretty big, it's not relatively bigger, but it has a lot of variants in it. So the first step that we do is we produce the bed file. We talked about how to produce bed files in demonstration number 1. And again, it's very straightforward using Flink. And we produce these bed files and they come with BIM and FAM files. So that gives us a good starting point for our further analysis. Then we look at the FAM file and we see that this particular individual has only one, the FAM file only has one individual in it. Next, we go to step number 2 of this notebook. So we will now process this file through VEP. It's a tool developed by Ensemble and it goes by the name of Variant Effect Predictor. What VEP essentially does is it goes into a VCF file and it annotates the VCF file variants. So as Professor Jordan talked about that, sure, there are a lot of variants existing on our chromosomes in our DNA, but not a lot of those variants are going to be pathogenic, or they're not entirely going to be deleterious. A lot of them, in fact, are benign in nature. And the idea is that we don't know what variant does exactly what when it comes to the whole molecular biology of the variant. So DNA encodes RNA and it encodes protein. So if there is a variant in DNA, it may or may not affect the protein, and it may or may not affect the protein-protein interactions happening downstream. So if it does not affect the protein-protein interactions, well, that variant is probably benign. So we use the tool VEP to know what these variants, what is their expected effect on if you're homozygous or heterozygous for these variants, what is the expected effect? So we look into the tool called VEP, Variant Effect Predictor Tool. Here they have really good documentation there, and they have a lot of ways to run this tool, a lot of data for that. But we are going to run it in a very straightforward way for our purpose. We're going to run VEP just using their cache memory. So they come up with a lot of databases. One of the key databases they come up with is ClinVar. And ClinVar, again, to follow up on what Professor Jordan said, it's again, it holds information about variants that are clinically significant. And it is, in fact, it's community-based, and it's each of that information that is annotated against every variant is, in fact, supported by different publications. So when we run VEP, so I would like to talk a little bit about VEP installation. So it comes in Perl programming language, and since new and modern systems do not have good support for Perl, I would recommend that if you install this tool, you install it through Conda package, and the details are there in our GitHub. But the installation could be a little problematic, but the tool is really good. So we run the tool on our patient and on their X chromosome, and we see what output VEP produces. So VEP only gives us 2 files. One is the output file and the other one is summary file. Summary file is an HTML file. We don't have to look at it. And we look at the output file primarily, and we want to see what data does it have. So the output file, it essentially looks something like what you see on your screen here. And the output file from VEP, VEP has successfully annotated a lot of variants. And if you go and look deeper into the file, here I'm only showing you first a few lines of the file, but we see the patient has almost 279,000 variants that have been annotated. So 279,000 variants from these files were annotated from this particular file. And again, we cannot go and look into 280,000, almost 280,000 variants and see which variant might be deleterious or which variant might be leading to the hemophilia condition of the patient. So what we do here is, and again, we're seeing intragenic variants. If you go down in the list, you will see something like an intronic variant, which explains by the name of it, it exists on introns. And sometimes they are in, the variants are contained in 3' region or 5' regions of the genes. So we're really looking to those particular variants which exist on the coding region or exonic regions, which will have, we're more certain that those variants might be playing some role in the disease that this patient have. So what we do here is we only look for missense variants. So missense variants are those variants that will, in addition to being on an exome or a coding region, they will cause a change in the amino acid configuration and the protein structure. And again, a lot of variants do not cause any variation in the protein structure. And if you recall the table shared by Professor Jordan, there are 64 possible combination of nucleotides, and they yield only 20 proteins. So you can have a codon and there's a change of nucleotide, but that might not affect the protein. And this is what we are trying to achieve in this notebook. We need to find that which of these variants, in fact, can be causing a problem. So we look for missense variants, and these are missense variants are often found to be pathogenic or deleterious in nature. So we try to find for those in this, among these 280,000, 279,000 variants. And here's a little bit of bash command that I've performed here. But what it's essentially doing is it's obtaining or extracting the missense variant. And after obtaining them, what it does is it goes to ClinVar. Again, Professor Jordan talked about it. I talked about it too. So ClinVar is going to annotate our variants, and it holds information through the support of community publications and various coming from various scientific journals. So when we obtain the missense variant for these particular individual, we see how many of those were in fact present in ClinVar. And we end up with only these 3 variants. So you see, we started with 279,000 variants, which could have a potential effect, but we end up with just 3 through this process, and we're only looking at those variants which are going to be directly affecting the protein structure and the downstream protein-protein interaction for this individual. And those variants, these 3 variants, could possibly be having an effect and leading to the patient's disease, which is hemophilia. So we will look at just one variant here, and now we have these variants. We're not sure that what it's causing. So we go on to internet again, because internet is very helpful, and we go to this really nice web tool called Varsome. So Varsome is a variant discovery tool, and it's an online tool. It summarizes the categorization, ACMG categorization for each variant. So Professor Jordan showed us multiple tables in which ACMG guidelines were shared. And these ACMG guidelines are, again, we won't go into the depth of them, but what they essentially tell us is, depending upon which guidelines are supported how much, we can comment on the pathogenicity or deleteriousness or benignness, in fact, of any variant. So we go to Varsome, and Varsome has different methods of taking in a variant. One of the easy method is, of course, you can just go in and give the RSID. But sometimes what you will observe is, in fact, in my practice as well, what I have seen is whenever you get declassified patient data in which patient identity is not there, the VCF files are not annotated properly. They just give us the raw information. So sometimes what you have is only chromosome position and the chromosome number. You do not have the RSID for those individuals. So what you do is you can create your own query for Varsome. It just, it's a very easy way to create the query. You give the chromosome number. In our case, it's X. You give the chromosome position, and then you give the alleles for this individual. So reference allele and alternative allele. And again, this information is not particular to this individual, but what is important here for us to understand is that this individual is homozygous for the alternate allele of this variant. So we put this query into Varsome, and it gives us really nice output. So this is a screenshot of what I obtained from Varsome. And what you see is I entered this particular variant information, and gives us the transcript ID or the gene ID of where this variant exists. And you see a lot of categorizations light up. And PM1, PM2, PP5, you can see these are the important ones. And overall verdict, it gives you as pathogenic. So we see that we are making some progress here, and we may be onto something. Varsome is saying that this variant, if this person, particular person, is homozygous or heterozygous for this particular variant, or like he or she has the alternative allele, they might be. This is like very highly likely to be pathogenic in nature. So we see that a lot of ACMG classifications light up. So Varsome also gives us a brief comment, also comments briefly on these ACMG classifications. And the most important ones here are PM1 and PP5. So PM1, it says it's a hot spot here, and it again comments on what I have said before about this, that this variant is on a coding region, and it affects the amino acid configuration of the protein, and it's likely 100% pathogenic. So it's very, very highly, like in my experience, there are very few variants that compared to the total variants we have, of course. There are not a lot of variants that come up as 100% pathogenic. So this variant, if somebody has it, it's very likely that some sort of disease, or in our case, hemophilia, might occur in the patient. And then we look at PP5. It says, again, it says that ClinVar classifies this variant. That is something we already know. But the next line, the second line here, is something very important, and which kind of concludes our whole notebook here. So PP5 here says that UniProt, which is a protein database, classifies this variant as pathogenic associated with hemophilia. And not only does it say that this is associated with hemophilia, it also gives us publication PubMed IDs. So these are PubMed IDs of publications that say that they have found, or they have scientific evidence to say that this variant is associated with hemophilia. So there are at least 15 publications here which say that. So what we observe here is that overall, that we started with the patient, just to summarize everything, we started with the patient's VCF file, and the patient unfortunately had a disease of hemophilia. So they were suffering from this disease, and we looked at only the chromosome X, and because we know hemophilia, hemophilia is related to, or a lot of variants on chromosome X are responsible for hemophilia. So we go in and we run the VEP tool to get the annotation. The idea here is to, again, bring the genotyping information to a level in which a human can interpret them. And that is one of the key areas in bioinformatics in general. We have lots of data. And one of the key responsibilities of bioinformatician is to bring the data down so that a medical professional or a doctor or a genetic counselor is able to convey an appropriate information, a relevant information, or a useful information to the patient. So we bring the data down to just 3 variants. From almost 300,000 variants, we bring it down to 3 variants. We go to Varsome. It's an online tool for variant discovery. We see what this variant might be associated with. So it's not, it's a faster way of going on PubMed and looking for different publications associated with this variant. Instead, Varsome gives the good summary of this, and through its ACMG classifications that have been defined, it gives us a good summary of what variant has what implications. And PP5 again tells us that we come full circle here, even though we knew hemophilia was there, but we can say with a little bit more of confidence. So we have confidence when we say that this variant might be a reason, or is probably a reason for the occurrence of hemophilia in this patient. So I think that concludes our demo number 4 for genomic diagnosis of rare disease. And I will, Professor Jordan, if you have any additions or any questions for this, we have, I think, at least 5 more minutes before the end of this demo. Any more questions? So I'll be happy to have them.
I have nothing more to add. That was very, very clear. Thank you, Shivam. Augusto, are there any questions for us before I move on to the next session?
No, la verdad no, perdón, la verdad hasta ahorita no tengo ningún reporte de alguna otra pregunta.
Okay. So let me share my screen. Can you stop sharing, Shivam?
Yes.
All right. Just a second. Okay. So now we're going to move on to, there we go. Now we're going to move on to a demonstration about how to calculate polygenic risk scores. So that last session was about rare variants, the discovery and the characterization and the filtering out of rare variants that cause Mendelian disease, right? So these are very low frequency variants that are at high penetrance. So these are causal variants. If you have one of those variants, you're very, very likely to have disease. On the very other end of the spectrum are variants that are found in high frequency in the population and have very low penetrance. So the fact that you have one of these variants doesn't mean you have disease. It just means you have a slightly elevated probability or chance of disease. And so what these polygenic conditions, so these are complex common diseases like type 2 diabetes, hypertension, various kinds of cancer, they are caused or they're associated with lots and lots of small, and by small I mean small effect size, variants spread all across the genome, all of which have small effect size. That means each of those individual variants only contributes a small elevation in the risk of disease. And a polygenic risk score is just reading across the genome and summing up all of the effects of those variants, each of which entails a small elevation in the risk of disease. So we'll see more about that. And you heard about this again in the talk of Claudia Latu. So genome-wide association studies, GWAS, can be used to discover genetic variants. So in this case, they are SNPs, they're polymorphisms because they're at relatively high frequency that are associated with, they're not causal, but they're associated with heritable traits of interest. And these could be diseases, that's what we're focused on today, but they can be also anthropometric traits, height, weight, body mass index, neurological traits, schizophrenia, personality, etc. And you can use these SNP discoveries, that is these SNPs that have been discovered through these very large GWAS studies, where they compare cases and controls to make predictions about the phenotypes of interest given the sequence of an individual. And that's exactly what we're going to show you how to do today. In order to be able to do that, you need to understand something about effect alleles and risk alleles, okay, that are discovered again in these genome-wide association studies. So genetic variants, alleles, these are the specific nucleotide variants at any particular position in the genome that are found at significantly different frequencies in cases versus controls are said to be associated with the trait or the disease of interest, okay? And the effect allele is operationally defined as the allele that is associated with the trait, okay? So if you want to build or use an application that predicts the probability of complex common traits from an individual genome sequence, what you're going to do is be counting the number of variants in any individual genome that are associated with disease and weighting the number of effect alleles. So the effect alleles are weighted by what's known as the effect size. And the effect size is just the probability that that particular allele, the level of risk elevation that entails that specific allele. So you're going to count all of the effect alleles in any given genome, and each one of those effect alleles is going to be weighted by its effect size. Again, the effect size is just how much elevated risk of disease that particular allele gives you, okay? So I just said that, but just to repeat, the strength or the magnitude of the effect or the risk alleles associated with the trait or the disease that you're looking at is measured as the effect size. And for binary traits, in other words, disease or no disease, we use the odds ratio. For continuous or quantitative traits like height, we use the slope of the regression, the beta value, between the allelic dosage for that SNP, how many alleles are there, and the trait measure, okay? So here's an example of GWAS summary statistics. And by now, this should be a little bit familiar to you because you've seen this a lot of time in Shivam's talks, right? So you have the location in the genome, the chromosome, the specific base pair in the genome, this SNP identifier here, and then you have these two different alleles. Now, in this case, they are simply operationally defining the effect allele as the first allele, A1. We have the frequency of the effect allele in the population, and this is the effect size, okay? So in this case, it's a beta value, so it's for a continuous trait, and this negative effect size means that the presence of this allele C is negatively associated with the magnitude of that trait. Here we have a positive effect size, so at this location, the presence of a C is positively associated with the magnitude of that particular trait. So these are the kind of summary statistics, and this is just the significance of those associations when you consider the effect size and the error. This is a point estimate of the effect size. Again, the magnitude, the slope of the relationship between the allelic dosage and the magnitude of the trait in the population of interest, the point estimate for that value of slope for the effect size, the error associated with that point estimate, and from the point estimate and the error of that estimate, we can get the p-value, okay? So you see that the association here is kind of weak. It's not a very significant p-value. This one is more significant, and this one is really, really statistically significant, right? So the probability of seeing this relationship, this magnitude of slope and the low error for this particular variant is highly significant. Okay, those are the summary statistics. And the way you calculate a polygenic risk score is you just read through that file of summary statistics and you calculate the weighted sum of the number of risk alleles found in the personal genome, right? So you have one patient, one person, you have their genome sequence, you've done a genome-wide association study, or someone else has done it on many, many thousands of genomes. And that study has identified all of these risk alleles. We go and we look at the genome of the patient or the participant in the study, and we ask how many risk alleles does that patient have? And it's a weighted sum. So the number of risk alleles is weighted by the effect size. That is the magnitude of the effect that each risk allele has on the trait for all of the alleles, okay? And so here's the formula for that. So let's say you have n number of positions in the genome that are associated with disease. Let's just say this is 1,000. So that you do a genome-wide association study, you find 1,000 different positions that are associated with disease. For each position from 1 to 1,000, you say, how many risk alleles do we have? And that can be 0, 1, or 2. You can have no risk alleles. I see you, Augusto, 1 risk allele or 2. You count that number and you multiply it by the effect size of the allele, and then you just sum that across all 1,000 variants. Augusto, pregunta.
Yeah, we have a question, King. So
Good.
the question is, let me just read it as it was written.
Okay.
Do this group of genetic diseases always happen in homozygous individuals?
No, absolutely not. So this is a really good question, right? So let me take one step back. So what is a genome-wide association study? And let's just focus on let's say hypertension, because it's a quantitative trait, right? So we say, and let's just do systolic blood pressure, okay? So a quantitative trait. So you take, let's say 1,000 people, and every single one of those individuals, you measure their systolic blood pressure, okay? So you have a quantitative health-related trait for all 1,000 people. And then for those 1,000 people, you characterize their genome over 1 million sites. And then you look at every single position that varies, and you relate the allelic dosage at that position to the systolic blood pressure, okay? So actually, this is a really good question, and we're doing fine on time. So if it's okay with you, Augusto, I'm going to try to get a plot that shows this. So give me 1 second.
That's okay. That's okay. We are okay on time.
Yeah, we're doing good on time. So I'm looking for a QTL. Sure. Here's an example. So all right, so let's imagine you have some quantitative trait, right? So let's just say this is systolic blood pressure on this y-axis, right? And you're looking at 1,000 people. And so every person can, at one position in the genome, they can have 2 A's, they can have an A and a G, or they can have a GG. And the slope, that beta value, the effect size, is the magnitude of the slope of the association of the effect allele with the magnitude of the trait, okay? And this is just one particular variant. Is that clear, Augusto? So every single variant is going to have a beta value.
Yes, it is.
Every single variant is going to have a beta value, which is going to be the magnitude of the slope showing the relationship between the quantitative trait, which in this case is systolic blood pressure, and the genotypes that are present at that particular location. And this is just one. This could be another one, but we'll have 1 million of those. Okay? And then what we often do is we say, okay, let's take the top 1,000 most significant ones. And then for all 1,000, we're going to read through the genome. So now let's go back to our score. So the effect size is that beta value, which is the slope, the magnitude of the relationship. This number is just how many effect alleles, right? So, okay, now back to the original question was, is it only homozygous? Well, no, it can be homozygous with 0 risk alleles. It can be heterozygous with 1 risk allele and 1 non-risk allele, or it can be homozygous for 2 risk alleles. And that can vary across all of the different sites. So we're looking at 1,000 different sites, and for every site we say, how many risk alleles do you have? 0, 1, or 2, and we multiply that number by the effect size for that site. Does that make sense? Augusto?
Yeah, it does. It does.
So this is just a weighted sum. It's a weighted sum of the number of risk alleles found in the genome. And remember, every risk allele increases your risk of disease just a little bit, okay? And we look at all of those all across the genome, and we calculate a polygenic risk score, okay? And the beautiful thing is that this information, this kind of work is happening all over the world in labs all around the world, and it's all being collected in this PGS catalog. So all of these polygenic scores, and at the time we built this, there were 829. There's probably a lot more. For 214 traits are all available on this website. And so what do I mean by available? I mean the summary statistics, right? The specific variants, the identity of the risk allele, and the effect size associated with that risk allele are available. And that's all you need to calculate a polygenic risk score. You take those summary statistics, the location of every variant that's associated with the disease, the identity of the risk allele that elevates the risk, and the effect size associated with that risk allele. That information across 10, 50, 100, or 1,000 variants is all you need to calculate a polygenic risk score from an individual's genome. Okay, so let me pause there and see if there's any more questions. And then we'll go into a specific case study, again, using Puente de Alto Costo, and Shivam will walk us through that. Anything, Augusto?
No, there are no any other questions.
Good. That was a great question. So back to Puente de Alto Costo. So rheumatoid arthritis is another disease that the Colombian government and their partners in this non-governmental organization have identified as having a maximum impact on the economy and public health in Colombia. This is a complex common disease, unlike hemophilia. So we're going to look at this as a case study. And as always, we're leaving you with additional resources if you want to learn more about this. So with that, Shivam, I'm going to turn it over to you to take them through the exercises.
All right. Let me share my screen. So thanks again, Professor Jordan, for giving a brief overview of polygenic risk scores. And just before we go into this particular notebook, let's try to get the essence of what we will be looking into. So as Professor Jordan mentioned, that we have a GWAS study conducted on rheumatoid arthritis. And GWAS study will, at the end, in essence, will give us the variants that are associated with this disease, as well as what is their relative contribution. That's one aspect of what a GWAS gives us. And this is the information that we will use to find a polygenic risk score. So polygenic risk score, let's say GWAS reports that we have a disease called rheumatoid arthritis, and GWAS, for example, this GWAS reports around 200 SNPs or 200 variants that are associated with this disease based on their cohort. So we can go into different individuals and try to see what is the variant, what is the genotype calls for these particular variants look like in these individuals. And in addition to that, we just can't consider every SNP as it is. We need to get the weightage of that SNP. And that is what polygenic risk scores is. A polygenic risk score is going to give us the relative susceptibility of any given individual to a disease based on GWAS statistic. And all this susceptibility is again normalized on a population level based on all the cohorts we process through our analysis. So let's start this notebook, and we will start by obtaining the PGS file from PGS catalog. So Professor John talked about rheumatoid arthritis, and we will obtain the file of ACPA positive rheumatoid arthritis from PGS catalog. You can just go there and it's fairly straightforward. You get the particular file. The link is there, and you can also fetch the link from here. You get the file. And next, let's talk a little bit about the cohort that we will be using. So we have the GWAS summary statistics that's coming from a group of individuals, but we are not looking at those group of individuals. Those were participants of that GWAS study. What we are going to look into is a new group of individuals for which we will try to find a polygenic risk score for the disease in focus. So a disease in focus is rheumatoid arthritis, and we want to see what a particular set of individuals, what the polygenic risk score for these individuals look like. So we get a cohort of Colombian population again from 1000 Genomes Project. This cohort has 94 participants. So we get the VCF file or the genotype data for all these individuals. One thing to keep in mind is that GWAS is usually performed on all chromosomes. And if you recall that we had been doing all our analysis on just chromosome 22, but we can no longer do that because, for example, this particular GWAS reported around 200 variants. And these variants, for example, you can see here, and we'll go into more depth there, but they're coming from chromosome 1, 2, 3, all the way to 22. So we have to get all the chromosome information this time for Colombian population. And we convert the VCF file to a BED file, BIM file, FAM file, like we talked about in all the previous notebooks. And now we get started with the real polygenic risk score calculations. So again, our cohort is Colombian population from 1000 genomes. There are 94 samples here. and we are using the GWAS summary statistics from a particular study on ACPR positive rheumatoid arthritis. So we're trying to combine these two information, and in the end, our ultimate aim is to get the polygenic risk score for these 94 Colombian individuals, which were participants of 1,000 Genome Project. So let's start with looking at the polygenic risk score, file that we obtained from PGS catalog. So this file is coming from a GWAS study that was conducted on rheumatoid arthritis, the disease in focus right now. And it has multiple columns, and it's if you remember from Professor Jordan's slide, it has multiple information such as p-values, what SNP ID it is, what that SNP ID corresponds to. For example, what is the chromosome number, what is the affect allele, what is the reference allele. There are three main columns. So let's look at what the information we need from this GWAS summary statistic. So we have a GWAS summary statistic here, and we will be focusing on three main columns, three main category of information. The number one is SNP ID, the RSID. This is a unique ID. We've talked about it multiple times. It corresponds to each SNP that exists that was there in the GWAS and will exist on the genotyping calls for Colombian populations. So there's a SNP ID, and then there's the affect allele. So this allele is the affect is the affect whose relation to the disease is being studied. So, for example, we look at a particular allele and we see what contribution of this allele was to this particular study. So similarly, we see we move on to the column number 3, which is affect weight or affect size. So the question is not only about what alleles are associated with the GWAS. The question also, the key question is also to look into what is the relative contribution of these respective alleles. So is that allele contributes a lot? Like what happens if you have two of those alleles? Like, do they contribute 50-50? Probably not. So we look into that, and these are the three columns that we have to keep in mind. So in this particular file, again, a little bit about the programming aspect of it. These files come in all sorts of formats. So you might want, if you're dealing with these files, you might want to go in and look at them a little bit. So for us, the RSIDs are in column number 1. The affect allele is in column number 4. So 1, 2, 3, 4. The affect allele. These are the affect allele that is in consideration, and it also has the column headed number 4. And then number 8 is the affect weight or the affect size. So what is the relative effect of this particular allele? So you see the different relative contributions of each allele. So for example, you see the last allele you see on your screen, RS1801274, has a greater effect than the first one you see, so 0.49 against 0.06. So now we move on to the next step, and we go to PLINK again for calculating our polygenic risk scores. And again, you can also calculate these scores using your own manual calculations. And if I recall correctly, Professor Jordan also had a slide representing the mathematical formula to calculate a polygenic risk score. But for simplicity and easy computation, we'll use PLINK for this particular processing. PLINK will go in and we'll talk a little bit about what exactly PLINK will look into. So PLINK needs this GWAS summary statistic file, so which has the RSID, the affect allele, and the affect size. And in addition to that, the PLINK also needs the column number of these three important columns. So remember, I told you that these come in different formats. So this is 148, column number 1, 4, and 8. It can vary depending upon what publication format was, or how the authors exported or supplied their file. So it changes, so something to keep in mind about that. And other than that, we have the VCF file or the BED file for all the chromosomes this time. Remember, these are coming from all the chromosome, not just chromosome 22. And this is purely because of the nature of the computation here. And these are coming from all chromosomes, and we have a BED file for them, and they represent the column. They're coming from the 94 participants of 1,000 Genomes project, and they're coming from the Colombian population. So we run PLINK here, and we ask it for calculate a polygenic risk score, and PLINK produces a file called .profile. And this file it simply gives us the final scores of our the final polygenic risk score for each individual. So in entirety, when we look at this file has 94 individuals and they all have their respective scores. So you can see scores are varying a little bit for all the individuals. And again, this is a particular disease of rheumatoid arthritis. Professor Jordan shared us results from, he showed a photograph or a Google image from high systolic blood pressure. And this is performed, I think, all over the world on different populations, different cohort for different types of disease, because as an individual, you really need to know what disease are you susceptible to. And this is something Professor Jordan mentions again and again, and this is something that the scientific community really supports, is that most of the diseases are a result, most of the diseases in any individual are a result of two things, genetic and environmental. They might be of different proportions, 50-50, 10-90, you don't know. But most of the diseases are a result of two things, environmental reasons and genetic reasons. So we really do need to know what is your susceptibility to a disease. So this is why we calculate the polygenic risk scores for these Colombian individuals. So now moving on, so let's move on to the next step of our notebook here. And this step particularly deals with looking at the ancestry of these polygenic risk scores. So recently, a lot of publications are coming in, a lot of research is being done, which looks for what is the polygenic risk scores for a particular disease in a particular population. So for example, African populations might have a slightly different or highly different polygenic risk scores to a particular disease when compared to European population. So this type of research is really gaining traction now. And this leaves us the question about, for example, the population which derive from two different clusters. So an example is Colombian population. As we observed in our third notebook, in fact, that Colombian population derives its ancestry from three different clusters: European, Native American, and African. So we're always interested in seeing that, depending upon the ancestry, what is the relative risk or polygenic risk of that population to a particular disease. And this is, again, very useful when it comes to the cost of sequencing. So we cannot go in and sequence every individual, but what we can do is sequence a particular region of a country where we know that the population or ancestry fractions are quite similar. So if a city has 1 million people, we cannot go and sequence everybody, but what we can do is get an estimate of what ancestry fractions look like in general for the city, and then find their polygenic risk score. So this is kind of what we're demonstrating here. So again, we go back to using our programming language. And the idea is to, we already have the polygenic risk scores. We need to know what the ancestry looks like for these polygenic risk scores. So once obtaining the scores, we bring in the ancestry information. So if you recall in notebook number 3, we derive the ancestry information for these Colombian individuals. So what you see here is two matrices. The first one gives you the ancestry fractions, and the second one gives you the polygenic risk scores. And again, what we're going to do next is we're going to merge these data sets by the IDs and merge these data set to get the scores and ancestry information together. And after that, what we're going to do is we're going to make a little plot. And this plot is, again, let me just make both the plots and then we can look at them simultaneously. So the first plot you see is has European ancestry fraction on x-axis. So as you go from left to right on the x-axis, the European ancestry fraction increases from 0% to all the way to 100%. And on the y-axis, we have is polygenic risk score. So as you go up in the y-axis, the polygenic risk score keeps on increasing. So what we see here is, for particular to European ancestry, and this is a line that just represents the general trend of individuals. The individuals are fairly well dispersed, and the polygenic risk scores does not change that much as the ancestry increases. So we can see that, as the population as the population ancestry fractions become more towards European, there's no change. in the polygenic risk score. But if you look closely at the other plot that we have, and again, this plot has polygenic risk scores on y-axis, but on the x-axis this time, we have Native American ancestry. And we see a little bit of increment in this as the Native American ancestry increases. So what the pattern, the slight hint of a pattern we are getting here is that as Native American ancestry is increasing, the polygenic risk score, remember, it's a susceptibility score to a particular disease based on your genetic variants. And we are looking at Colombian individuals here who were sequenced in 1,000 genomes. So the risk of these individuals increases, slightly increases as their Native American ancestry increases. And again, this is I would say, a demonstration example. A lot of research is being done for heart diseases, systolic blood pressure, diabetes, kidney diseases. So this notebook is meant to demonstrate how we calculate polygenic risk scores and how we can see the relation of polygenic risk scores to ancestries. And that sums up the notebook. And I think we have 5 more minutes left. Professor Jordan, if you have any additions, any questions, that'll be nice. We can move accordingly.
I don't, Shivam. That was very, very clear. Thank you for that. Augusto, do you have any questions there in the chat?
Not so far. We don't have any additional questions. Let me just check our Excel. So that might be really interesting if I mean, the audience can formulate some questions. Please go ahead, ask, examples, clear ideas. I don't know. We have time. We're pretty much on time. Is this the last? No, we still have one.
We have one more session, so we're right on time.
Good, good. That's okay. Okay. Seems we don't have any additional questions, so we can move ahead then.
Okay, I'm going to share my screen and give the last set of introductory slides. So now we're changing gears. So just again to recap, in order to implement precision public health in Colombia, we need a deep understanding between genetic ancestry, genetic diversity, and health in the country. So we started off with the first 3 sessions on first understanding how genetic diversity and genetic variants are encoded so we can analyze them bioinformatically. Then we did a review of genetic ancestry inference techniques. And from there, we transitioned into the health side of things, where we looked at rare diseases caused by highly penetrant variants. And then we looked at complex common diseases, rare diseases caused by one or very few highly penetrant rare variants. And then we just did polygenic risk scores for complex common disease caused by many common variants with low penetrance, each of which elevates your risk of disease just a little bit. The last thing we want to talk about with respect to the relationship between genetic variation and human health is pharmacogenomics. The science of how is it that your genetic variants influence how you respond to different medicines, okay? So this is a very interesting case and one of the only cases where there's a relationship between common variants that also have high penetrance. So in my talk, I showed that pharmacogenetic variants are largely found at relatively high frequency within and between populations, but they also tend to be protein coding sequence variants that are highly penetrant. We're not sure why that's the case, but in this spectrum, we're at this part here, which is very unusual for disease, but actually not unusual for pharmacogenomic variants. So variants that have high penetrance and also have relatively high frequency in the population. Okay, so just a quick review. I talked about this morning, but for pharmacogenomics, many medicines are currently prescribed as though they work equally well in all people. So the dose of the medicine is adjusted depending on the age, size, and gender of the patient. However, even if patients have the same age, size, or gender, they might still have different responses to the same medicine because of genetic variation, okay? So as a result of this, the choice of medication, which medication to give, or the dosage of any particular medication can be tailored to an individual patient by analyzing their genome sequence, thereby personalizing their treatment. Okay, so pharmacogenomics is the study of the relationship between genetic variations and how our body responds to medication. And pharmacogenomic testing gives us insight into how any individual patient can respond to a drug. So when we think about the different dimensions or the different aspects or the different ways in which genetics can influence how we respond to drugs, we think about toxicity, whether or not a medication will induce an adverse reaction in a patient, whether or not a medication will make some patients sick and others not, based on their genes, their genetic variants. Dosage, how much of a medication should be given to a patient based on their genetic variants. Remember from my talk, simvastatin toxicity is common in the Colombian population of Antioquia. It's associated with European ancestry. Whereas in the population of Chocó, tacrolimus, an immunosuppressive drug, which is used in organ transplants and for individuals that have severe allergies, there's an allele that is common in Chocó and associated with African ancestry that is associated with higher metabolism of the drug, therefore necessitating a higher dosage. And then there's efficacy, just how well will the drug work. So PharmGKB is the pharmacogenomics knowledge database. It was developed by Stanford University with funding from the National Institutes of Health. It's a partner of the NIH-funded Pharmacogenomics Research Network. And the idea is to help researchers understand how genetic variation among individuals contributes to differences in the way that those individuals react to drugs or medications. So for the PharmGKB, there are 4 levels of evidence, and within levels 1 and 2, there are different divisions that correspond to the strength of evidence for the relationship or the association between any given genetic variant and response to a drug, okay? And level 1 is high, level 2 is moderate, 3 is low, and 4 is preliminary. And it's only level 1A, so there's not that many variants that are officially recommended clinically. The other ones are more under research, under investigation right now. So this, I think, is very, very simple and doesn't require a lot of introduction. Pharmacogenomics is the relationship between genetic variants and how people respond to drugs with respect to toxicity, the dosage of the drug, or the efficacy. Pharmacogenomic variants are stored, annotated, and distributed by the Pharmacogenomics Knowledge Database, PharmGKB database, and they're given different levels of evidence, of confidence rather, with respect to the evidence in support of those associations. So if you want to understand more, here are some resources. And with that, I will turn it over to Shivam for the very last session. In this last session, we're going to take a genome sequence of an individual patient, a Colombian or a group of Colombians, and ask about their predicted response to a specific drug based on the presence of a particular pharmacogenomic variant. So Shivam, I'll turn it over to you there. And Augusto, if there are any questions at any time, please stop us. Shivam, it's all yours.
All right. Sorry. Thank you so much, Professor Jordan. Thanks again. Let me share my screen. All right, so as Professor Jordan gave us a brief introduction about pharmacogenomics, I can briefly talk about what we are looking at here and how we're going to do it. So we're going to start with a drug called methotrexate. It's usually used for treating cancer, and it's used as chemotherapy. And again, this drug is, we're interested in seeing what is the relative toxicity of this drug and in relation to what the genetic ancestry of people look like. So as Professor Jordan mentioned, that drug dosages are, always tuned according to age, sex, and other physical attributes. But there's not many recommendations about how to tune them according to genetic variants. And one of the big things is that different genetic variants or different genetic ancestry might change your body's response to specific drugs. And that's what PharmGKB is tries to capture as an information. It reports the toxicity of particular drugs in different based on different genetic ancestry. So we will again start this notebook with some data. So the first step is to look at the data we have. Again, we are interested in Colombian individuals, which were participants of 1,000 Genome Project. And these are 94 individuals that we have. We have genotyping performed on them for all chromosomes. And one of the genes that we are interested in is MTHFR. So this particular gene is, we want to focus on the variants that lie within this gene. So the first thing to do is to extract only the variants that lie on this particular gene. This gene lies on chromosome number 1. It's a fairly large chromosome. So as a bioinformatician, I am always, tempted to not run it on the whole VCF file. We are only going to tune it down to the variants that exist between these two positions. These two positions are again obtained through NCBI. One of the things that you have to, and this is, I should have talked about this in the first notebook, but one of the big things as a bioinformatician you have to take care of the genome build. So genome reference that we have in our scientific community was updated multiple times, and the two popular ones are GRCH37 and 38. So I am using GRCH38 in this analysis. So if you end up using GRCH37, all your calculations will be off. So I suggest not using 37 for this particular analysis. So depending upon your data, you might be using 37 or 38, but the key thing here is that always make sure that you're using the right genome build. So we look at these positions. We gain these positions from NCBI for this particular gene, which exists on chromosome 1. We have the VCF file for chromosome 1 for Colombian individuals or participants in 1,000 Genome Project. The first thing we do, as I talked about, was we will extract only the variants that are present on this particular gene. So doing that with Plink is again relatively easy, just a command away. So you use the command extract, and you use the argument range, and you give it the position ranges here. So your position ranges.txt file is here, and we're giving that particular file here. So it's going to extract all the variants that exists on chromosome 1 between these two positions. So once we have that with us, we will again recode our data into a VCF file this time. So we perform all our analysis on bed files, as I talked about before, and bed files are binary representation in this whole genotyping data. It's always nice you can always convert bed files back to VCF files. So we perform that particular operation here. And as I initially described or talked about, we will be looking at this particular variant on this gene. So we will filter out the data for this variant, and we will see what the genotype calls for this particular variant look like. So we have, again, just to recap, we have 94 Colombian individuals who participated in 1,000 Genomes Project, and we want to see whether they have the alternative allele, whether they have the reference allele, and what is their if they are homozygous or heterozygous for that allele. Corresponding to this particular SNP on which resides on this gene on chromosome 1. So let's look at a VCF file here. We look at we have the 94 Colombian individuals for this particular variant, RS 180113 or 1133. And we have 94 Colombian individuals here, and here's their genotype call for that particular SNP. So you can see that the first individual, you can see the ID here, it's 8G01112. That individual is homozygous for reference allele, so it has AA there. And there's quite a bit of heterogeneity here. You can see there's some individuals also are homozygous for the alternate allele, so they have GG here, and a lot of them in fact have AG here. So 01, it corresponds to AG. We do not know about parent 1 and 2, so we cannot say GA with confidence, so the data is not phased, but we know that they're heterozygous for AG. So The next thing we do is we go to Farm GKB, which Professor Jordan talked about. It's developed by Stanford University through the with the contributions of NIH. And we search for this particular variant, 1801133, and it again resides on the MTHFR gene. So what we find here on Farm GKB is that this variant has a toxicity relationship with the drug called methotrexate. So again, methotrexate is used in cancer, so and it's used as a chemotherapy agent. In fact, if you see here, it's a chemotherapy and immunosuppressive drug. So again, toxicity for chemotherapy is something very important, and we need to be absolutely sure of the dosage of this particular drug, and that is why this particular thing that we are demonstrating here is important. So this drug we find the relationship of this particular SNP with this drug, and here you can see the level of evidence. So this is 2A, so we would say it's moderate according to the classifications. And if you remember, as the level goes into 1A or 1B, 1A is in official guidelines. So 2A suggests that there is some evidence, at least, for the toxicity of this particular drug, methotrexate, against this SNP. And let's look into what alleles cause the toxicity. So, this SNP can either be encoded as AA, AG, or GG in the individuals or the participants who are getting genotyped. So we read here, and what we find is that AA is associated with increased risk of toxicity. So it says it may have individuals who are AA in genotype and arthritis who are treated with methotrexate have an increased risk of toxicity. So again, this drug is also used for arthritis, and for those particular individuals, if you have an AA allele, and if you're homozygous for A, you have an increased risk of toxicity. And if you look at GG, it says you have a decreased risk of toxicity. So this brings us a very important point, and just to summarize what we're doing here is, depending upon this variant, you might be looking at increased or decreased toxicity risk, and depending upon what allele you have, whether you have the reference allele or the alternative allele, your dosage, if more scientific research is put into this, your dosage could be tuned upon the allele you have for this particular variant. And finally, we look at the heterozygous AG allele, and it says that it may have an increased risk, but further evidence is required. So essentially what we see is like AA and GG, there's a big difference. One gives you increased risk of toxicity, and the other one gives you a decreased risk of toxicity. And which takes us to the next step of our exploration on Farm GKB. So this is again on Farm GKB, developed at Stanford. So the next thing we look at is we look at the allele frequencies for this particular variant that we have been talking about. So what we see here is something very interesting. And again, we haven't analyzed anything in a Colombian population yet. We're just simply looking at this particular variant. And what we observe here is that this variant A, or the reference allele A, is very highly prevalent in the Latino populations or the populations of South America, as well as in the Ashkenazi Jew population. So you see 44% prevalence and 46%. So what you're seeing here is like AA will give you a high risk of toxicity, and this AA is relatively more prevalent in the Latino population and the Ashkenazi Jew population. And you can see it's relatively lower for other populations. For example, the Finnish population has a lower prevalence of this allele. So, and again, this gives us a direction of what the research we're trying to do here, or what our ultimate aim is, to understand if certain populations might be, depending upon the population's ancestry fractions, if these populations might be more susceptible to toxicity for the particular drug. And again, this can be done for other individuals and other drugs as well. So you observe that this allele A is more prevalent, and now we go back to our R notebook. So we will do some analysis in which we would like to see, let me remind you that we started with Colombian population VCF data, and we brought the data down to a particular SNP on a particular gene particular SNP or a particular gene. And what we will do is we will go to RStudio and we will import this data. So first of all, what we'll obtain is we'll get the ancestry data. We have been using this data for in almost all our notebooks. We produce this particular matrix here in notebook number 3. And then what we do here is we're obtaining those Colombian individuals who were participants of 1,000 genomes. These individuals are homozygous for the reference allele. That means that these individuals have AA for that SNP. So they have an increased risk of toxicity. And what we do here is we just simply write these individual names, and we just subset these individuals out. And the idea here is to look at their ancestry makeup. So we again perform the same thing we performed in notebook number 3. We just manipulate and transform our data frames to produce an admixture plot for these individuals. So let's talk about the individuals. These individuals are a subset of Colombian participants of 1,000 genomes, and they were homozygous for AA, reference allele, for the SNP that we have been talking about. So these individuals are again have a higher risk of toxicity if they have been prescribed methotrexate. And this is what their ancestry looks like. Again, this is just to see if they had a higher percentage of a particular ancestry against something else. So we can see a little bit higher percentage of Native American ancestry, but again, we cannot be sure about this. This graph doesn't tell that exactly. So we move on to another type of representation for this information. So here we will be using a library called, in R, it's called VCF, VCFR. And what it lets us is, it lets us read the VCF files into our R data frame. So we read the data frame, and what we are doing is we're encoding our information for 00 at reference is AA, 01 is AG, and 11 is GG. And we annotate that information. We also try to find the ancestry and combine the ancestry data for these individuals. Again, this data comes from all the 94 individuals, and we will drop them for only the individuals that are, we're looking at all 96 individuals here. And now what we are going to do is we're going to plot a particular graph for the toxicity to the drug methotrexate. And this is based on only the SNP RS180-1133 that we have been talking about throughout this notebook. And what we see here is we have 3 sections, or three little graphs in this whole image. And the first one corresponds to the European ancestry fraction. So what you see is on the y-axis, you have European ancestry fraction. So as you go from bottom to up on y-axis, your European ancestry increases. Similarly for the middle, the blue one, the African ancestry increases. And similarly for the red one, the third, the Native American ancestry increases as you go from bottom to top. So y-axis gives you the ancestry fraction. And on the x-axis, if you look closely, I can try zooming in. So if you look closely on x-axis, it's divided into, for each individual graph, it's divided into 3 ticks. The first one is for AA, the second one is AG, and the third one is GG. Again, remember, if you have the AA, or if you're homozygous for reference allele A, you have a higher risk of toxicity. And what we observe here is that so what we observe is, like, as you go from AA to GG, the European ancestry fraction is decreasing. And again, as you go, as you have the AA have the highest risk of toxicity. And for the Native American, we have a similar figure, which shows us that as you go from AA to GG, that means as you're going from higher risk of toxicity to lower risk of toxicity, your Native American ancestry decreases. Similarly, as you go from AA to GG in European, so AA is highly toxic, GG is low toxic. So as you go there, the European ancestry is decreasing. So what these graphs are, again, these are a way of representing this information that we have. And what this notebook helps us to perform is it's going to be really helpful for public health policies. depending upon what genotyping information we have for a particular population. So this is a demonstration of how you can use the genotyping data to get an idea about the toxicity of a specific drug. And again, this can be scaled for multiple variants, multiple populations, and accordingly, the public health officials can work on it. So I think that concludes our demonstration number 6. And Professor Jordan, if you have any questions, any additions, that'll be really nice.
I don't have any questions, Shivam. You've done a great job. Thank you very much. Augusto, we have come to the end of the practical session. We've got a few minutes left. If you want to, if there are any questions, or if you or Leonardo want to give any closing remarks.
Great job, Shivam.
Thank you.
I don't know what's going on with my camera, but it should be working. Anyways, what I would like to say is thanks to you both. Thank you, Leo, Shivan. That was a great job. So far, we don't have any additional questions. Just let me check the Excel. Oh yeah, there is another one here. Jorge Mario Vasquez is asking if the pharmacogenomics study. Let me read in Spanish, and then we'll translate. So he's asking how to assess the effect of variance given the ancestry percentage in pharmacogenomics.
This is a great question, and I'll take that. So the thing about the pharmacogenomic variance, so if we actually, you know what, I'm going to open up my slides, and I'm going to go back to the second slide that I showed. So I hope they'll be able to see this. Okay, so here, so remember that these pharmacogenomic variants are quite unique in the sense that they're both found at high frequency in populations, and they have high penetrance. So this is very different than the Mendelian disease variants that are found at very low frequency but have high penetrance, and the genome-wide association study discovered variants that we use for polygenic risk scores that are found at high frequency but have low penetrance. What does this mean? This means that high penetrance means that if you have this variant, you are very likely to have the phenotypic effect associated with the variant. Now, for Mendelian variants, that phenotypic effect is disease, but for pharmacogenomic variants, that phenotypic effect is the response to the medication with respect to toxicity, dosage, or efficacy. Now, what does this mean with respect to ancestry? And it turns out that most of these variants are protein coding variants, and they're non-synonymous variants that change the encoded protein. Now, what this means with respect to different ancestries is that for the most part, there could be exceptions, but for the most part, these pharmacogenomic variants will have similar effects on the individuals in whose genomes they are found, irrespective of that individual's ancestry. So the presence of a specific pharmacogenetic variant is highly indicative of the phenotype, the specific phenotype that it's associated with respect to drug response. So the effects of the variants are similar across ancestries. Now, what can and does really differ is the frequency of those variants in the populations. So remember from the talk I gave this morning, the frequencies of the variants are associated with genetic ancestry. And if genetic ancestry differs between populations, as it does for Antioquia and Chocó, for example, then you will expect many more people in one or the other population to have the response to the drug that is associated with that variant, right? So there's kind of two different ideas, and I just want to summarize those. So on the one hand, the effect of the variant itself is not really affected by the ancestry, but on the other hand, the frequency of that variant in different populations is very strongly tied to the ancestry. I hope that helps, and I don't know if you want to add anything to that, Leo or Shivam.
No, I think that explains it very well. Yeah.
Good. So there is another question. Let me just read it. Janice Valencia is asking, could one gene have more than one pharmacogenomic variant for a specific drug?
Yes, absolutely. That's a very good question. And a lot of these pharmacogenomic variants. So remember, we talked about the concept of linkage disequilibrium. So if you think about, how genes and chromosomes are inherited, you inherit an entire haploid chromosome from your parents. Now from one of your parents, right? One each from each parent. Now that long haploid chromosome is broken down by recombination over generations, but SNPs that are in close proximity in the same gene are inherited together over many, many variations, or over many generations, right? So you can have multiple variants that are linked, all of which are associated with a particular response to drug. And it turns out that a number of these pharmacogenetic tests test what they call haplotypes rather than the specific individual variants. So the haplotypes are just combinations of different variants. So that's one thing. So multiple variants in the same gene can be associated with the same response to a drug because all of those variants are linked and inherited together over many generations. On the other hand, it turns out that many of the genes that are implicated in the response to drugs come from the same family of enzymes. These are cytochrome P450 enzymes. So these are enzymes that are involved in the metabolism of lots of different products that we consume, not just drugs, but also food and lots of xenobiotics. So different variants in different cytochrome P450 genes can be associated with different drug responses. So the answer to your question is yes and yes. Multiple variants in the same gene can be associated with the same response to drugs, and different variants in the same or different genes can also be associated with different responses to different drugs. Good question.
So, thank you, King. So I want to capitalize on that. So from a practical point of view, I mean, some of these variants might be just linked, okay? So assessing all of them might not be worth. Maybe just one that would be indicating that perhaps the other one is already present, right? Is already there. Okay, good, good.
Yep, absolutely.
Good. So we have another question. Hold on just a second. Marisol Aguirre Rojas is asking, how do you calculate the dose for the medicine according to the patient pharmacogenomic profile?
Right. So now, in order to really be able, so remember that lots of these variants are still under research. So remember the different levels of confidence, 1, 2, 3, 4, and then within 1 and 2, it's only 1A where there are specific recommendations. So this is a very active area of research, and there's only a few drugs for which there are very specific recommendations in terms of how you calculate the dosage based on the variant. Warfarin is a great example of that. And Shivam, can you do some Googling for me while I answer this question? Look up warfarin dosage calculator and see if you can find something. So remember, we talked about the dosage of a drug depends on your age, your weight, and your sex, male or female, but also your genetic variants. So warfarin, a drug that's commonly used, a blood thinner, there's a lot of information on that, and there are studies that have related specific genetic variants to the rate of metabolism. So that's the connection to dosage, right? You take this drug and your body starts trying to break it down to metabolize it right away. If you are a slow metabolizer, if you break the drug down slowly, you'll need a lower dose of the drug. Whereas if you're a fast metabolizer, if you break down the drug quickly, you need a higher dosage. So remember the example of tacrolimus in Choco, the T allele, which is associated with African ancestry, is associated with more rapid metabolism of tacrolimus. So in order for the immunosuppressive effects of tacrolimus to take hold in patients that have 1 or 2 of those T alleles, they'll need a higher dosage. So For the few drugs like warfarin, where we have a lot of studies, really solid evidence on the relationship between the genetic variant or variants and the dosage, there are these calculators whereby you plug in the information with respect to the variant, the other information about the patient, and it gives you a formula in terms of dosage. So Shivam, show us what you found. You're muted. You're muted, Shivam.
Shivam, you're muted.
Yeah.
Can you see my screen? I'm sharing it. Let me zoom in.
Yes, it looks good.
Yeah, so just to talk about, this is warfarin dosing calculator. So I was able to find two calculators, and this one has more information. So it asks for a race, and it gives all the options there. And the other calculator asks whether you specify a race as non-black or black. So I'm not sure about the ancestry, how it takes or how the dosing varies. But again, for calculating the dosage, it's asking the patient's race. And I think that has a little bit to do with the correlation of the self-reported race with the ancestry of an individual. And maybe Professor Jordan can talk more about it.
Shashwat, I like the other calculator better because it has the genetic information. It doesn't look like this one has genetic information. So those variants, so let me just kind of explain this. So we've been talking in terms of individual SNPs, single nucleotide variants, but those historically were actually characterized as parts of so-called haplotypes. So remember, a haplotype is a single chromosomal region that encodes multiple genetic variants that are inherited together. And those haplotypes, what you see in the genetic information are the names of those haplotypes. So that's actually an older, slightly lower resolution technology. But this is great because we had a couple questions ago, or one question ago, where do multiple variants in the same gene affect dosage? And that's definitely the case for warfarin. So you would do one or more of these different tests, and then you would put in this other information about the patient, their age, their sex, their ethnicity. I do want to point out that Shivam said, can you go to the ethnicity again?
Yes.
Shivam said it includes all of the options.
Yeah, so I don't know if you can see my screen right now, but it asks for race as well as for the variant.
Just to be clear, so this is for the United States, right? So obviously race and ethnicity are social constructs which are defined very differently for different countries. So the meaning of race and ethnicity, or ethnia, would be very different in the United States than it would be in Colombia. And that underscores the need for country-specific solutions to this. Anyway, that's sort of an example of the answer to your question. Now, there's not a lot of cases of drugs where we have this much information, but this is how you would include that information in the dosing recommendation. And again, at the biochemical level, and I want to be really clear about this, the relationship between genetic variants and dosage has to do with the rate at which drugs are metabolized. So if a genetic variant confers a more rapid rate of metabolism, so the protein is able to more rapidly metabolize the drug, the patient will need a higher dosage. And conversely, if a genetic variant confers a lower rate of metabolism, you need a lower dosage. You don't want to give them too much so they overdose, so they'll need a lower dosage. So that's how that information is included. And this page that Shivam is showing is a very nice example of how genetic information is integrated with other information from patients to make the best possible recommendation. Now, please remember that warfarin is probably the single drug for which we have the most pharmacogenomic and other information. But as these studies continue coming in, there'll be more and more of these recommendations, more and more of these variants that rise up to that 1 or 1A level, whereby they get encoded into the medical recommendations.
Thank you, Shivam. Thank you, King. Just want to add something.
Also, thanks for that question. Both questions were really great and helped us to
nice.
elaborate on this.
So just want to add something, and that, I mean, warfarin is a perfect example from the point of view that the therapeutic range is very narrow.
You're
so
still
you
sharing,
are not allowed to do any mistake. So that's why this kind of things depends really in every specific case for every specific drug. And that's why we're calling the attention on studies like this should be done specifically having into account not only the genetics, but also the ancestry, because it affects the therapeutic range of all of these medications. Really nice question. Good. Let me check. There are no any other questions here. So I think we're pretty close done. Should we just say goodbye and leave it there?
Are we going to have a clausura?
Oh, just a few words, that's all.
Yes, Augusto, I want to thank you very much for the opportunity. And I also want to recognize Shivam and Shashwat, who's not with us today, that just did a tremendous amount of work in putting together the exercise sessions over the last 2 weeks. And Shivam that did a great job delivering them. So very well done, Shivam. And a big thank you to you and also to Shashwat.
Ok, muchísimas gracias. Entonces voy a devolverme al español y simplemente quisiera, en nombre de todas las instituciones involucradas y los profesores que participaron en esta cátedra, darles las gracias por haber asistido. Esperamos que pronto la participación de manera sincrónica sea muchísimo más. Este material va a quedar disponible en una página web que vamos a dejar como memoria con cada una de las conferencias que fueron impartidas el día de hoy y con cada uno de los ejercicios que también hicimos. Muchísimas gracias por estar presentes hasta este momento. Un profundo agradecimiento a la Comisión Fulbright Colombia por su apoyo en la realización de esta cátedra. Y bueno, gracias a todos y hasta una próxima oportunidad. Que estén muy bien.